Okay, good morning, everybody. My name is Oscar Lopez. I'm a director of the University of Pittsburgh ADRC. It's still very unfortunate that we are meeting in a hybrid mode. I hope that next time we can do it in person for everybody. I would like to thank Dr. Silverberg, Elliot, and Townley for providing the framework for this meeting. And also, I would like to thank the NACC team for the wonderful organization. This is going to be a very special meeting. We'll be honoring John Trojanowski this morning. As you know, he passed on February 8th of this year. He was a giant in the field of neurodegenerative disorders and mentored the careers of many of the new generations of leaders in neuropathology. Dr. Edward Lee and David Walk from UPenn will provide us with a more personal view of his scientific achievements and impact in the field. And I thank Dr. Eddie for the article that you wrote in Acta Neuropathologica, which is excellent. We'll initiate the morning with an NIA update from Dr. Hodes, Masliah, and Silverberg. Dr. Sarah Biber will provide us with a NACC update. Dr. Jeff Dage, an update from NCRAD. We will announce the 2 Rising Star awardees from the REC and the 3 NACC New Investigators Awards. Dr. Catherine Possin from UCSF will give us a summary of the ADRD summit. Then the scientific presentations will be initiated by Dr. Pete Nelson from University of Kentucky. He will talk about the contributions of John to late. Adam Boxer from UCSF will be presenting the contributions of John as a pioneer of tau therapeutics. And Dr. Charis Wisdom from UCSD will talk about fluid biomarkers in Alzheimer's disease and other neurodegenerative disorders. So we will start with the first speaker, and I think it's Dr. Hodes, which will be a virtual presentation.
Good morning. Thank you for the opportunity to be here with you. I hope both video and audio are coming through all right. It's a pleasure to participate in this discussion and open with some updates from NIA about the work and goals we have in common, progress over these past years. The next slide, please. Begin with an update in appropriations and spending and what's new since the last meeting with you. Next slide, please. So this year in FY22, we had our budget earlier in the spring, relatively late in the fiscal year. NIH has a total appropriation of $45 billion. For NIA, the budget increased to some $4.2 billion, which now leaves NIA as the third largest of institutes at NIH, in large measure due to the targeting for research in Alzheimer's and related dementias. This year, among targeted areas for the appropriation, some $289 million additional for AD and ADRD. Some others which are relevant to our common interests as well: targeted increase in Down syndrome, in brain, and in all of us. Next slide, please. So this is an idea of the year-over-year increase in NIA appropriations from a little over $1 billion to now some $4.2 billion. Next slide, please. And as noted, a great deal of this is due to the year-upon-year increases in targeted resources for Alzheimer's related dementias research, beginning at the largest scale in 2016, and for each year through the present. This year, as noted, another $289 million. Next slide, please. Among the strategies we've taken to be best stewards of this research have been to make it available to promising projects from across all of NIH. And this particular program, now in its 5th year, is one in which we are providing supplements for AD, ADRD related research to grants which themselves are not AD focused and which are funded by any of the institutes at NIH. So you can see here in this slide for FY21, nearly $90 million was allocated to these supplements across nearly all of the institutes and centers. Some important and innovative research, as you can imagine, NINDS the largest, not a surprise, but NHLBI, for example, reflecting some of the cardiovascular contributors and correlates of the dementias. NIBIB, with contributions to a great deal of the technologies and technologic forefront capacities to the field. And so this continues to bring new talent, new perspectives into the field of aging research. Next slide, please. One reflection of the status right now, important to all of us, is the ability to achieve pay lines for Alzheimer's-related dementias research. And illustrated here are the current allocations. The year's not over, and it still may change in the positive direction as the year goes on. But you note for the AD, ADRD, CSR-reviewed percentile applications, the pay line, as it was last year, is the 28th percentile, with increases for both new investigators and early stage investigators to the 31st and 33rd percentiles accordingly. And you can note that for the general pay line, not AD and ADRD relevant, these pay lines are substantially lower as they have been at the 10th percentile, for example, for the less expensive applications, with 13th and 15th there as well for the early stage investigators. Next slide, please. For the non-percentile, those NIA reviewed applications, you can see it for AD, ADRD, for program projects and other NIA reviewed, 40 priority score, career development, 35, and fellowships. And here you may note, although the general pay lines are lower, we've done our very best here too in career development and fellowships to maintain support for people entering the field at the stage of developing their careers. Next, please. Just a note, from 2015 through 2021, even as we've been able to increase the resources for those already in the field of Alzheimer's-related research, about a third of NIA Alzheimer's disease-related dementias awardees were either new or early stage investigators, and about a fourth fell in the category of new to the field. These are individuals who had never before even applied for NIH support in this field. So bringing new perspectives, novel ideas into the field has been a part of the growth of our research investment. Next slide, please. Some updates at NIA. Next. So one metric of the outcomes of all of the basic and translational research that happens is, of course, the number of active clinical trials. Here's a summary in the next slide or two of NIA AD, ADRD clinical trials. So for pharmacologic interventions, some 8 at the late stage, phase 2-3 or phase 3, but notably now some over 60 early stage trials targeting 14 different targets. And this is the result of early translational work that's now coming into the clinic and will provide the input to the later stage definitive and pivotal pharmacologic trials. In addition, an even larger number of non-pharmacologic interventions targeting exercise, cognitive training, combinations of these areas. Next, please. Dementia care and caregiving, important in a large number of trials targeted at the gaps in knowledge there, so they can quickly be translated to those currently living with dementia and those who are care partners with them. And other trials in areas such as diagnostics, biomarkers, and the treatment of neuropsychiatric symptoms that accompany AD and related dementias. Next, please. I want to share with you here a report that came from the National Academy's Decadal Survey of Behavioral and Social Sciences, which was examining the impact of social science, behavioral sciences on reducing impact of dementia in America, and points to a number of priorities that would be pursued actively, looking at cause and solutions, notably for disparities among populations in the risk and progression of Alzheimer's-related dementias. Next, please. Another area that we're focusing on is that of small business. I think many of you will be aware, SBIR, STTR, as mechanisms for encouraging the translation of research and research involving private sector or private-public partnerships. The challenge here is one in which a boot camp is provided for applicants, providing entrepreneurial expertise, mentoring, all with the goal of bringing a new, expanded, and diverse population of individuals into small business-funded research and accompanying the academics and translating the fruits of basic research into all aspects here with private sector participation. Next slide, please. And finally, it's a privilege to be the first to note the memorial to John Trojanowski, who this meeting is and so much of what we do. When I first joined NIA at a personal level, John was among the first to offer himself as a valued mentor and friend. His counsel was invaluable for NIA. He served on our board of scientific counselors, on our National Advisory Council, And of course, in this group, he was always an active part. I would say, again, at a personal level, when I presented before all of you, the one person I could count on for questions and probing comments was John Trojanowski. And so we can just take a moment to pause for the first of many occasions in memoriam for John, and with best wishes to his legacy, to his family, and to all who have learned and profited so much from John. And next, please, just a offering again for all of you to please stay close to and informed with us in the future years through these links or through contact with myself. All of us are here privileged to be able to work with you in our common interests. So I turn it back to the moderator and on with the agenda.
All right, good morning. I'm Eliezer Masliah, and I'm the director of the Division of Neuroscience, and I'm going to give you an update on some of the research and funding opportunities that we've been working on this year. It has been an incredibly busy year for us, and this is going to be kind of like a fast review of a lot of what has been happening in the last few months. Before I start, I also want to thank all of you for being here, to the organizers for putting together really an amazing program, and all the challenges of doing this in a hybrid manner. This is the first live meeting for me, so it's quite interesting. And I also, before starting, just want to recognize once more the tremendous influence of John Trojanowski. I mean, he was always a fantastic collaborator and friend to me, incredibly generous and collaborative and friendly. And if Virginia is attending the meeting virtually, I want to send my warmest regards and support to her for all the amazing things that they have done to the field. All right, so we've been, of course, incredibly interesting in supporting understanding Alzheimer's disease in the context of the interactions between aging, genetics, and the environment. I mean, these have been major areas of focus on the research that we've been supporting well, for the last few years, but also in the last few months. And how does this have an impact on understanding neurodegeneration, developing new diagnostic, and developing new treatments? And in the area of genetics, as you know, what we've been doing, it's increasing the ability of the Alzheimer's disease sequencing program to expand to studies of diverse populations. The plan is to have over 100,000 whole genomes by 2025. And as you all know, there has been a tremendous progress on the Alzheimer's sequencing in the last few years. Over 75 risk loss identify, specifically 20 genes definitively assigned to risk to Alzheimer's disease. And now the ADSP on the NIGA side has put together a really beautiful summary of all these research that I really encourage you to take a look at. Also complementing all what this infrastructure of the ADSP has been done, now we have a new consortia on artificial intelligence, machine learning, on harmonization, and functional genomics. And this functional genomics consortia that just got started has at its main role to understand the impact, the functional impact of the genetics on ADRD, and really to enable discovery toward prevention, diagnostic, and therapeutics. This consortia has an external advisory, an executive committee, and an NIA leadership program that is directing a number of subcomponents. We have at least 6 groups integrating this program that include a number of different interesting aspects, including single-cell technologies, inflammation and microglia, diverse model systems, functional genomics across different networks, and the idea is that all these data will become publicly available, as with others, through ADSP, NIGADs, and the Functional Genomic Consortia. Also, along the lines of genetics, and in addition to functional genomics now, we've been putting a lot of emphasis on transposable elements. This is something that we're really extremely interested, both from the point of view of genetics, aging, DNA damage, infectious agents, and so on. And just recently, also a new one on non-coding RNAs. And our idea is that all of this is hopefully going to lead to a genomic medicine approach to AD. Now, a lot of these genetics of Alzheimer's disease have really contributed to the idea that in addition to the traditional pathways that we're already aware, like APP, presenilin, endocytosis is really a major pathway represented by the genetics, as it is lipid metabolism, neuroinflammation, and immune response, where there has been a tremendous amount of work been done and supported, and as we could expect, synaptic and neuronal signaling as it relates to tau. But endocytosis is especially interesting because how it links to lysosomal activity, and I think in this regard there has been tremendous excitement in the last few weeks because of the discoveries with the TMEM106 protein involving endosomal lysosomal processes that for which polymorphisms and mutations have been described a number of years ago on FTD and more recently on DLB and other neurodegenerative disorders. And now with the identification of the fibrils of TME106B by cryo-electron microscopy and the understanding of the structure. And of course, the Alzheimer's centers have been absolutely crucial for all the genetic studies that I mentioned, for many of the funding opportunities that we're supporting, and of course for all the research that has been done on the cryo-EM of all the fibrils in AD and ADRD, including TME106B. This is the study of David Eisenberg, but also there have been other studies where Indiana University have been supporting a lot of that research coming from Michel Goddard, and a lot of really fascinating work from the group at the Mayo Clinic and other areas, where they have identified these fibrils not only in FTD, but also in DLB and PSP. And what is more relevant, these fibrils have also been identified in normal aging and even in Alzheimer's disease. And apparently by cryo-EM, these fibrils have some diversity and might explain the differential susceptibility in these disorders. However, I know we always get very excited when a new amyloid is identified in these neurodegenerative disorders, and I know we have amyloidogenic pathways for all these different proteins, and now we're identifying many of these proteins in aging and Alzheimer's disease, and really we need to ask the question, well, what is truly the role of amyloid pathways in neurodegenerative disorders to the extent of they play a role in gain of toxicity, ER stress, etc. But also that these proteins could go through other pathways, which are the non-amyloidogenic pathways. There are a lot of C-terminal cleavage of these proteins that could be affecting function, having off-target effects, disturbing lysosomal function, and so on, that might also be responsible for a lot of the pathology. So I think you'll see in the next few years that we're also going to be emphasizing the idea of not only focusing on the amyloid pathway, but also try to focus on other pathways related to these proteinopathies. And I think the concept is that probably upstream many of these proteinopathies, both in amyloidogenic and non-amyloidogenic pathways, might be affecting endolysosomal function that could lead to protein aggregation and neurodegeneration, but also it's possible that independent of the protein aggregation, this endosomal lysosomal function might be leading to neurodegeneration through other off-target type of pathways. So I think we really need to think much broader about these disorders. And I know that there is always the concept that maybe protein aggregation is just a byproduct. I know we have been having a controversy about this for many years, John playing a major role on this, but I think it's something that we should continue to think about. Now, in terms of aging and mechanisms of aging, in particular, we have talked about proteostasis, lysosomal function, but other mechanisms, including DNA damage, self-senescence, and immune surveillance, are important. I have talked to you about them before, and this quarter, we're supporting a number of new aspects of aging in AD and ADRD, including mechanisms of brain hypoperfusion, lipid in brain aging and AD-ADRD, the impact of liquid-liquid phase separation, which is a really hot topic in the field right now, and I think it's something that we really want to encourage further research. The idea of the association between neuronal vulnerability and proteinopathy is also a pretty hot topic for us, as well as the idea of connectivity in the brain and how this connectivity is being disrupted with aging and AD, and T cells. I think we have focused a lot on microglia. I think focusing more on T cells would also be very fruitful on understanding and mechanisms of neurodegeneration. And just very recently, we had a workshop on lipids in brain aging and AD. Some of you were, and again, many of the ADRCs have been major contributors to this. And this was part of the funding opportunity that we put forward. I mean, there was a lot of very exciting research presented, including, for example, this one where again, this is a non-amyloidogenic pathway where, for example, aging induces the release of myelin and debris of myelin that are lipid-rich that produce a microglial response that is TREM2 dependent, not ApoE dependent, and that this white matter damage, microglial response, inflammatory response, is earlier actually than the DAM response. So a lot of very exciting research coming up. Now, the other area where also we're putting a lot of emphasis right now, and I know it's all over the media, it's everywhere you hear, is the exposome and the environment, not only toxins, pollutions, but also environmental factors, trauma, social, behavioral aspects, and so on. And for example, in the area of infectious agents, we've been supporting a lot of research, both on COVID as well as in other infectious agents in Alzheimer's disease, on the microbiome, but in particular, the exposome as a whole. And an interesting thing that came up in the recent appropriation that Dr. Hodes was talking about is an additional 15 million for exposome centers, that because where we are right now, well, you hear on our council just approved a NOC that we have on exposome centers. This is going to be administrative supplements. You're going to see that NOC coming up very soon. I mean, we've been funding a lot of research in addition to these. And as I mentioned, we had also a workshop about exposome. And when we refer to exposome, I don't only refer to the traditional environmental factors, but like physical chemicals, but also ecosystems, lifestyle, and social factors. And obviously, this has a tremendous impact on another topic that has been central to everything that we're doing right now, which is this topic of diversity. Diversity is really penetrating throughout absolutely everything that we're doing, and this is having a major impact on our focus, all the way from training to support on different grants through supplements, data sharing, and so on. And you're going to see a lot more coming up in the next few years on these. We just recently had this funding opportunity on bilingualism, cognitive reserve and resilience that also includes this idea of diversity. I'm going to talk to you just a little bit about this opportunity coming up on precision medicine approaches, minority health and disparities. We have a health disparities opportunity. Also screening for cognitive impairment, major emphasis on diversity, and these collaborative studies for which the ADRCs are perfect, and all the infrastructure that we're supporting are perfect for this opportunity, and with the idea of diversity being a major aspect. So as I said, we have recently this workshop on bilingualism, cognitive reserve and resilience, and again, thank you to all of you that participated. We're a number of gap and opportunities were identified, including improvement of measurements of bilingualism and diverse populations, establishing new multidisciplinary collaborations, and developing and testing new theoretical frameworks for bilingualism, both as it relates to reserve and resilience. And was some really very exciting research presenting, including, for example, this work where, for example, we could see that the effect of bilingualism in ADRD is more in the age of onset of the disease rather than in the incidence. And again, the white matter, the topic that I was talking about before in terms of lipids and inflammation, well, we see that white matter changes in bilingual individuals had an important reflection on reserve and resilience toward neurodegenerative disorders. A lot of really fascinating work going on there. As I mentioned before, another area, and this is something that is going to come up very soon, is this idea of building infrastructure for precision medicine approaches for minority health and health disparities. This is a collaboration among different divisions at NIA, and I mean, we do really recognize the need to capture neuropathological processes along environment, sociocultural behavior, and other demographic factors. I know a lot of you here are doing absolutely fantastic work along those lines, and I mean, we want to further support this type of research and create more infrastructure, as well as pilot projects and multidisciplinary teams. Damali Martin at our division is heading this program, and along with Nina Silverberg and Cerise Elliott, and of course many of the other programs that I mentioned before are supported by them as well. I also want to mention this recent project that NINDS funded with the ADRD funds, where Charlie DeCarli and Muriel Fomage are the PIs, which is a diverse vascular contributions to cognitive impairment and dementia. And again, with this big emphasis on the diversity now on vascular disease, vascular dementia, over 750 African Americans, 750 Hispanic, and also Caucasian. So really a very diverse program among many different entities. And of course, Charlie's here, and I'm sure he'll be happy to talk more to you about this program that we're just kicking out with NINDS. And we also have Keith here from NINDS, if you're interested to talk to him. Just, yeah, there you go. He'll be delighted to meet you and talk to you. And just finally, to close, I wanted to talk to you about some of the efforts that we've been doing to further bolster clinical trials, clinical studies, and drug development in Alzheimer's disease and related dementias. As you know, we have a very robust program that Larry Ruffolo, Susanna, Zane, Yin Yuan, and Christina McLinden, along with Lori, Ryan, and others, have been supporting, and we really have programs along all the different aspects of the drug development program, all the way to clinical trials. But there is two areas in particular in this pathway that we want to reinforce. One is this drug discovery for novel AD/ADRD targets. Again, we've been funding amyloid tau, inflammation, neurogenesis, synapses, and so on for some time, and Dr. Hodes has showed to you all the diverse targets and early stages that we're funding, but we really want to fund more of the new targets in AD/ADRD, and we just recently reissued the AD drug development program that has been an incredibly successful program. Over 10 drugs now have gone to IND stage. So this drug discovery for novel targets, the idea is really to bridge between what we're doing with AMP-AD, with TREAT-AD, and Model AD, all these new targets that have been discovered by all these different programs and tested in animal models, and now in a pilot early stage, the assays and so on being developed, now have a bridge between those programs and the drug development programs and the clinical trial funding opportunities. So that's our new drug development program. It's going to be an R61, R33, which is a new or relatively new mechanism for this process. But the idea is to expand our portfolio on e-therapeutics toward precision medicine and new drugs. And there is already some interesting publications coming here. This is actually a preprint. This is from bioarchives, but they have screened a number of drugs already, over 171 compounds that could be distributed and shared with a number of people in the community. These compounds are already known in the field, have been QC'd, and could be used to interrogate the validity of a number of emerging models, developing assays, and so on. So really a lot of great resources to the community are being developed. And I know a number of you here are part of this program, so really very excited about this. I also want to remind you, NINDS also have been putting forward a lot of new opportunities, both in the areas of early stage therapeutics for AD/ADRD, for disparities that we've been talking about, for treatment of Lewy body dementias and frontotemporal degeneration, which were areas where we didn't have targeted programs for drug development and therapeutics, as well as functional validation of novel targets, similar to what we're doing with AD for COVID and so on. And again, you can talk more to Keith or to Rod Corrible. They'll be happy to talk to you more about these programs. So the idea is that all these new infrastructure programs and opportunities that we've been developing along genomics, phenotypics, functional genomics, machine learning, leading to data harmonization. So the disease heterogeneity and diverse populations will include it, and we can move forward faster toward drug discovery, toward personalized medicine, given how close we are to 2025. I mean, it's right around the corner. And of course, I'm here and happy to talk to you during the course of the meeting about any questions you have. I think Lori is also here, and I see Christina, and Cerise, and a number of other people from NIA. I saw Don Rowe, Melinda Kelly. So we're really delighted to see you in person and chat about any questions, topics, or things we could be of help. Those that are from NIA that I didn't mention or didn't see, I apologize, but you'll identify yourself. Thank you.
Next speaker is Nina. She will be virtual.
Everyone, I hear an echo. Oh, I guess it's gone. I apologize for not being there. Wish I could be, and I'm grateful to my colleagues who went in person, and I hope that you all have an opportunity to meet and talk to them if you haven't met them before and see them in person. I also want to thank Dr. Hodes and Dr. Masliah. Those were great introductory remarks about what's so much that's going on. I'm going to also start and mention Dr. Trojanowski. He, of course, had a big influence on me as well when I first started. And one of the many things that he shared with me and the community was a major analysis of the network of Alzheimer's centers and how we all interact as a community. And I think he played a major role in fostering that network and the community of the Alzheimer's centers specifically, as well as including ADRD research, particularly Parkinson's. He kept encouraging me strongly to include Parkinson's and Lewy body and other types of dementia in the center's program. And I take that to heart, and I feel that that is one of the major unique aspects of our Alzheimer's Center's program. And so I think everything on this slide you all know, but I just kind of wanted to remind our community of the central role that the ADRCs really play in global, not only Alzheimer's research, even though we're called Alzheimer's centers. I think it's very apparent what role the centers play in the related dementias as well. And the fact that we have all of these included, I think, has enabled us to really be leading the field scientifically and developing new approaches and improving our understanding of the fact that Alzheimer's is certainly not a unified entity. So we also provide standardized data, samples, and participants across the different dementias and severities. We are able to sustain support with strong community ties, which really translates into better retention of the participants and involvement of the participants. Training the next generation in a multidisciplinary environment is a key to the center's program. And another unique aspect is the autopsy services that support not only the participants and their families, but major research advancements. And the fact that the center's program is part of a larger national infrastructure, including NACC, NCRAD, SCAN now, NIAGADS, and AGSP, and others contribute to many of our national and international signature programs, NIA funded as well as others, NIH funded. So I think key to this is acting both within the network and all of our interactions across centers, as well as locally. And I mean both the public local community as well as the local research community. We do need to improve our sharing and open science practices because we have so much in the program. I think it's incumbent on us to really do a better job, even more. I know we're doing a lot, but to improve the sharing that we've been doing. I've had people approach me from outside the center's program many times and have trouble accessing resources. And I know that some of you have talked to other entities like the Gates Ventures folks and others who are looking to help with this. So it's been identified as a need from outside. And I think we need to work together to figure out the best ways to improve on that. Just as another example, I know that many of you were part of the ADRD Summit, as Eliezer mentioned, many of these big meetings having anything to do with Alzheimer's. The centers are very, very well represented. So I think we're playing a major role in advancing the science throughout the field, and I want to recognize and appreciate that. I have now 3 slides in here that I've included on NACC. I just want to emphasize how much NACC has really stepped up. I hope that you've all had a chance to meet some of their new staff. You've heard from Sarah, Biber, and Carrie Stevens, and Sean Mooney are all there, and many others. I don't want to start naming people because I'll leave some out. But I really do hope that you'll have an opportunity to see all of the many, many, many different activities that they've been involved in and ramping up. Expanding data coordination. I know Sarah went over this slide yesterday, and so I'm not going to go into the details on this one, but I just want to recognize that not only have they expanded their data coordination role, but really stepping up and more to come on coordinating across the network, improving communication, working with our steering committees, particularly working on the UDS version 4. There's a lot coming through on that. This pipeline database, interfaces, imaging as SCAN ramps up. There's a lot going on with respect to NACC and bringing in the imaging and connecting both the legacy that you heard about yesterday and the future imaging that will be coming. I'm going to just briefly mention the COVID-19 supplement that NACC has and how that's going to impact hopefully our future interactions with NACC, and then some digital biomarker activities. So this is what they envision in terms of pipeline and database, the interactions with the ADRCs, SCAN, NCRAD, ADGC, and NIAGADs. So we have, the imaging data, biospecimen and samples, genetic data, and this is going to help with our interconnection and availability, open science, open data. And then you see here the current work that's going on with longitudinal clinical data, neuropath data, legacy, imaging data, and planning for digital biomarker, digital neuropath, and electronic health record and CMS data. So lots of activity going on. This is a schematic of some of the activities involved with this COVID-19 supplement. NACC is currently working on a pilot with 3 of our centers, Indiana, Columbia, and WashU, to get COVID data from the electronic health record and bring it to connect it with the NACC data with Medicare claims data. And hopefully this is going to help us. We have some of the deepest phenotyping of cognitive data, and we hope to learn more about the impacts of COVID on older adults and particularly people with dementia. But the idea here is that this will be a trial of bringing in electronic health records and CMS data to connect with the center data. So trying it with those 3 and hopefully expanding to others. I want to also give kudos to NCRAD. NCRAD has also been going through a very rapid expansion, doing a whole lot more than they used to. And some of the areas that they've expanded, they have a large number of iPSCs, fibroblasts, and brain tissue now available for distribution. The online catalogs are available for both iPSCs and NCRAD Family Study brain tissue. Catalogs for fibroblasts and the Gift of Life study are coming soon. And under the NCRAD umbrella, not only do they bank Alzheimer's studies, but I hope you're all aware that they also have FTD, REM sleep behavior disorder, Down syndrome, aging studies. So in total, there are 60 studies with over 100,000 participants. And the Alzheimer's centers are one of those 60 studies. So if you have ADRD collections that you would like to bank with NCRAD, please reach out. We want to make sure that you're aware with respect to the fluid biomarker initiative. NCRAD accepts plasma, Buffy coat, and PBMCs from the Alzheimer's centers. They provide the kits and the shipping. They return ApoE, GWAS data, and now plasma biomarkers for A-beta 4042, p-tau 181, and NFL for free for participating. So please send all of your participants, and they are looking for more. I want to, again, because this has been ongoing and people have been working very, very hard, I'm going to thank again the clinical task force. You see the names here. We want to provide special thanks for the support of Hannah Kraft for the cognitive working group, and to Spencer Lowe for the technology group that is really, really ramping up now. And the social determinants of health working group led by Lisa Barnes at Rush and Megan Zielsdorf at, oops, what just happened? Wisconsin, I think I went backwards. There we go. And you will be hearing soon about the outcomes from the social determinants of health working group. We have a new behavior working group that is added on to the clinical measures group to help address behavior, mild behavior, and how we should be measuring it in the centers. So as I discussed last time, there's a lot of interest and excitement about what we can do with digital technology. I think many, if not all of the centers are using some digital tools already. We're taking a two-pronged approach to bring digital across all the centers together. So the first part is the tech working group has identified a tech UDS 4.0, which is comprised of tests that have been developed in academic research centers, including from some of our ADRCs, from the UCSF ADRC, courtesy of Kate Possin, the TabCat, which you're going to be hearing about, has cognitive tests from Emory, courtesy of Alan Levy's group. There's VisMet, which is an eye tracking task. And from OHSU, courtesy of Jeff Kay and his team, there's CART. And all these tests are going to be delivered using a tablet and can be administered through your research clinic. So we're starting with in-person tests to ensure that it can be done with everyone that you have currently enrolled. And we'll be testing a standard e-consent that has already been used by the Wisconsin and Emory ADRCs. This will set the stage for not only obtaining electronic consent during the center visit, but also for later on during for remote assessment. And they'll be using the Linus Health platform to help integrate all these different applications into one research dashboard. At the technology session later today, you can learn more about the specifics of the tech UDS and the pilot study plans that NACC has to develop the infrastructure support for digital data processing and storage. Second, because so many ADRCs are already using digital, we want to generate an inventory to capture what everyone is doing. So the BU ADRC, and thank you, Spencer Lowe again, is helping to conduct one-on-one interviews with each ADRC to gather this information and put it into a knowledge portal so that you can all see what everyone's doing. And hopefully this will help also spur collaborations between centers. And as part of the interview, they're going to be trying to understand what you would like to be doing as well, and what would be needed at your center to make it happen. So NACC is going to be sending out a short survey, keep your eye out, to collect the contact information so they know the right person at each of the ADRCs for these interviews. And here's the information. I hope that those of you there in person and those of you online will be able to attend the digital biomarker CTF session later today. So another topic area that there's been some activity on is about ADRD data and indigenous nation sovereignty. And this is at the crossroads between two movements coming from NIH and the scientific field in general. We wanted to, of course, focus on big data, which is a little bit in conflict with the idea of recognizing the sovereignty and indigenous nations' right to control their data. And so we have started, facilitated by Carrie Gleason at Wisconsin, an indigenous-led initiative to provide guidance on use of these data. And you see the two contacts there, Lauren McCluster Davis from the United Nations in Wisconsin and Jordan Lewis from the native village of Naknek. And the contact information is on this slide, which will be available later. They wanted to start talking with us at NIA, and we've talked to other NIH level folks at the Tribal Health Research Office to work on a plan for what to do with native data to assure that there is data sovereignty. And potential stakeholders are encouraged to reach out to these leaders. And the Tribal Health Research Office from NIH has a tribal consultation on draft policy for data management and sharing. I have a link here, you can look it up. And also for the ADRCs, we have this Native American Interest Group listserv. And I hope that any of you working in Native populations, if you're not already on that listserv, will join. There's another at the top here, I have on the top right, the RADX program, which is for Rapid Acceleration of Diagnostics, has a separate tribal data repository. So you might want to take a look at that as an example of the type of way that this might work out. We have been working very hard. I have a whole little section here. You know, I've been working with the Neuropath Corps for a long time to help raise awareness of the need for brain donation. And we've identified multiple areas of need, specifically primarily diverse populations and control brains that are needed. So we've done a lot of work with the Office of Communication and Public Liaison at NIA to get the word out. The bottom right has an article that was in the Atlantic in 2018, which was really great. I was interviewed as well as other NIH folks and Neurobiobank folks. I think Howie Rosen was also interviewed in that one. But we're really trying to get the word out about the need and what neuropathology really contributes to the field. And so one of the activities that we've done is thank you, Dirk Keen, who's led this effort working with Neurobiobank. We started a pilot to have the Brain Donor Project, which is the referral source for Neurobiobank. They're referring to a limited radius for 3 of our sites, University of Washington, University of Pittsburgh, and Yale University. So thank you to Julia Koffler and Anita Huffner from Yale as well. Successful in integrating ADRC and Neurobiobank. We've had lots of interactions across the Neurobiobank activities and our ADRC pilot sites. We worked with them to develop a triage plan because they were getting a lot of both brain donor offers for Alzheimer's as well as scientists requesting tissue for Alzheimer's research. So the triage plan emphasizes the needs that I mentioned before, clinical trial participants as well as others. We have had a number of successful referrals, but brains have been collected. This has been going on for a couple of years now. They've been able to collect a data set on those brains that have been collected, including toxicology, which Neurobiobank does, but many of our sites don't do, and have begun uploading data to Neurobiobank. And the next phase will be sending samples to requesters. So you can see that this is going to be a benefit to having the central site, and we hope it'll be helpful with increasing autopsy with available data. But this is different from what we have at the centers. And so we're trying to understand what might work best to go through Neurobiobank and what might work best to go through the centers, and making all of this available to researchers and increasing the donor pool. So we'll update more in the fall at the Neuropath Core meeting and hope to have more to say about this Neurobiobank pilot. I hope many of you attended the late meeting in February. I think you may have seen the Alts Forum did a 3-part series writing up the summary of the meeting. It was fantastic. There's an NIA website that still has the meeting. You can go watch it. You can watch the associated posters that were virtual video presentations. And I think we're learning a lot. There's a lot of exciting science going on. Nobody's trying to say that this is a new disease, but it has been newly described, and it really has, just recognizing it, I think, has really contributed new research and new areas to look into. I also want to draw your attention to, there's many webinars on the NACC website that I hope you'll go back and take a look at if you missed them. There's also a YouTube channel. But thank you to Maggie Flanagan, who has organized the Digital Neuropath webinar series. Lots going on in Digital Neuropath at our centers, and she has really taken the time to organize a fantastic webinar series. So I hope you'll have a chance to take a look at those. The ORCOR also has been active, and most recent webinar was just in April. Gina Green Harris from Wisconsin gave a fantastic, so fantastic that she's been asked to do a part 2 webinar on participant advisory boards. It was really, really great. So I hope if you missed that, you'll go back and take a look at that one as well. We also have been working behind the scenes. Thank you to Krista Mulder for organizing all this from WashU. ADRC best practices. You can see this is from the NACC website. Some are already there and some are coming soon. The Administrator Steering Committee is working on External Advisory Board best practices document because the one that we had from before was pre-pandemic and a lot of things have changed. So they'll be working on that. We want to make sure that the genomic data sharing policy information is available there as well. And then a new area that we're going to be talking about is risk and biomarker results disclosure. This is definitely going to be changing very rapidly, but we'll be working with the Agreed Dementia group, which I have another slide on, to develop these disclosure best practices. Wanted to also make sure everyone's aware that we have an NIA-funded Research Centers Collaborative Network, which is collaboration across all of the centers. And all of the NIA-funded centers, not just the Alzheimer's centers. So they have a bibliometric tool that they've developed to allow searchers to identify which NIA centers are working in what areas. So I hope you'll all check that out at the RCCN website. It uses funding acknowledgements and keywords from PubMed to allow searchers to see which centers are publishing in what specific areas using a graphical interface. And they've been having workshops. These are supposed to interconnect among multiple of the centers, maybe not necessarily all of them, but NIA has many centers programs, and we'd like to be able to facilitate interactions, help, people at some of the Pepper centers and other the RICMARs identify which Alzheimer's centers might have expertise that they might be interested in working on. They have mentees as well. So I think it would be helpful if you all check this out and help facilitate cross-center interactions. Another activity that some of you may not be aware of is this group called GEAR 2.0, which stands for Geriatric Emergency Care Applied Research Network. This is being led by Eula Wang and Manish Shah. Eula's at Yale and Manish is at Wisconsin. They've identified a major gap in research. We have very little research on emergency medical care for people with cognitive impairment or dementia. And so they have engaged with a wide variety of stakeholders, with emphasis including people with dementia and their care partners. And this was a R61, R33. So they have identified and prioritized research gaps. And are planning to support and grow researchers to improve emergency care for people who have cognitive impairment or ADRD. And I will say that this has gotten certainly more relevant with COVID. I personally have been in the emergency room, not for myself, but where there were other people with dementia in the emergency room. And because they have not been able to have their care partner with them during COVID, it has been a major problem. So I've been glad to be involved with this. They really just wanted to raise awareness. I can't go through all of what they've already done, but they just want to make sure that you're aware, and I hope that you will check out their website and consider interacting with this group. The Agreed Dementia, which I mentioned earlier, is being led by Neelam Agarwal at Rush, Carrie Gleason at Wisconsin, and Allison Rosen at Stanford. They have gotten these 3 aims together: risk evidence evaluation of science, guiding when and how to provide effective risk evidence education, consultation to support ethical and legal risk mitigation, because they have lawyers and genetic counselors and all kinds of people involved with this group. There are these 6 subgroups of the Agreed Dementia group. Again, I don't have time now to read through all of their major accomplishments, but this has certainly become front and center with new FDA approvals of medication. We really need to be able to help people decide whether they want to have biomarkers evaluated and disclosed, and what the best ways to disclose those biomarker results are. And so again, I encourage you to check out the website, agreeddementia.org. And all of these subgroups are open. I think the information is available on the website of how to get involved. And the group meets monthly as a whole. And then each of the subgroups, I think, are also meeting monthly individually. We just wanted to be sure that you're all aware this is a reissue. This came partly from our review of the center's program, where we were encouraged to provide opportunities for collaboration across the Alzheimer's centers. This is open. It's not just the Alzheimer's centers. You can collaborate with Pepper Centers or any other RICMAR, other groups. It doesn't have to be a center even. But Cerise Elliott is the contact for this notice, and it's associated with PAR 22093. And we just really wanted to encourage collaboration, especially since NACC used to have the collaborative grants. This is sort of replacing and expanding upon those opportunities. And last 3 slides are from our Communication and Public Liaison Office at NIA. They wanted you to be aware that they'll be highlighting Alzheimer's and Brain Awareness Month this June. They're hosting the Alt Science Twitter chat on June 15th, which will touch on Alzheimer's research advancements over the past 10 years and exciting things to look forward to in the future. So several ADRCs have already let them know that they're participating. So if you want to participate, please reach out to Reese Western at NIA. We'll also be sharing resources and materials on Alzheimer's throughout the entirety of the month on Facebook and Twitter. And we also wanted to make you aware that alzheimers.gov, the federal portal for resources on Alzheimer's and related dementias, is now also available in Spanish. So please check that out and share with folks. And last but not least, new NIA publications and videos. Forgetfulness, Normal or Not, is a video, an animated version of NIA's popular infographic on this topic. Inside the Brain article on the role of neuropathology, and thank you again to our centers contributing to that. And you can find lots more health information and news on these different channels of the NIA website. And a reminder, as always, please, if you have papers coming out that will be of interest to the press and the public, we want you to contact the NIA press team. You can copy me and or Cerise, but ideally directly the NIA press team, and please provide them a lay level summary of why your work is so exciting and should be of interest. And that is all I have.
We have time for questions. We have a lot of time for questions. So I have a question regarding funding and the cost of grants that will be affected by the cost of living and inflation. Do you expect there will be more investigators asking permission to submit grants above the 500,000 K?
Richard, I'm hoping you're going to answer that.
Yeah, it's a good question. Don't know to have the answer. Sorry, I hear an echo. Okay, better. Yes, we're certainly tracking this carefully and prepared to see. The average cost, as you know, of research in general has been going up, both due to the inflationary cost as well as the increased complexity of a lot of the research we do. But fortunately, the resources have allowed us to keep up with this over past years. So what we do encourage is that, of course, that budgets, when they're proposed, are done thoughtfully and carefully, but that they are adequate and sufficient to carry out the mission. And with some of the emphasis we're putting on such things as diversity and recruitment into clinical research and trials, data sharing, we recognize that all these come with increased costs, and we encourage and are receptive to those costs being included so that we can fully meet those goals and targets. There are other complexities, maybe the question not exactly asked, we're seeing around COVID. And here we're responding to exigencies as they arise, to increase in costs, delays, but we're responsive case by case and encourage people to keep in touch with program staff over any of those sources or reasons for increased costs and expenses.
Okay, thank you. Any questions?
We have one from the chat from Robert Vassar. Does NCRAD measure A beta 4240, p-tau 181, and NFL in plasma? What platform is used for these measurements?
I was going to cover that in my session here in a minute, but yeah, we do measure p-tau, A beta 4240, neurofilament light. The p-tau 181 is on the version 2 Quanterix Simoa platform. Neurofilaments on the Quanterix Simoa platform. Euroimmune is what we're using for A beta 4240. And we are partnering also with C2N for people who are interested in Persivity.
I think that was Jeff Dage for those of us on the video. We can't see the audience, but thank you, Jeff.
Okay, since we don't have more questions, Dr. Biber is going to provide an update from the NACC, but she will be introduced by Bud Kukull. But are you there?
I'm here. I'm ready to go. First, I'd like to thank all the ADRCs and thank NIA for all of this great work that's going on. I'm always really impressed to see the summary of everything that's going on. It's truly amazing. And then I also wanted to mention or give a shout out to your NACC team and all the great work they've done to provide you with this hybrid session, the first hybrid ADRC directors meeting that's been done, basically, and a lot of work has gone into that. Now, before I move on, I wanted to introduce Sarah Biber, who joined NACC as the program director this past November, and she's trained as a PhD scientist, but she really has been helping us with her experience in leading operations for complex centers and programs, and has really organized much of what we're doing and some of what Nina mentioned in her slides. So now Sarah can continue from here and show you a lot of the things that are going on within NACC that'll lead us into even more exciting things as we move on into the coming years. Thanks.
Thanks, Bud, for the kind introduction, and we miss you. Sorry you're not here with us in person. So I'm thrilled to be here with all of you and honored to have the opportunity to speak on behalf of the NACC team. Before I get started with the talk, I wanted to mention that everything, as Bud said, I'm going to share with you today has been made possible by the incredible team at NACC. Can all of the NACC team members actually that are in the audience, many of you, some of them may be in the hall, please raise your hand. Okay, everyone look around and see. There's many more helping virtually right now. Yeah. Yeah. Yeah, and it's an incredible team. I encourage attendees to connect with them and learn about the great work that they're all doing. Let's see if these slides advance for me. There we go. There's the team. Okay, so the uniform data set has had a big impact on the field, and we're eager to collaborate with all of you to grow this data set and further leverage it to advance the field. At present, the UDS data set includes more than 45,000 participants with a median of 3 clinical assessments per participant and a max of 12. This is an incredible longitudinal data set. It also includes more than 6,980 neuropathology data sets from 58% of deceased participants. To date, more than 900 studies have been published using NACC data, and there are currently, as you know, 37 Alzheimer's disease centers across 26 states that are contributing to growing this data set further. As I'll explain during my talk, at NACC, we are on a mission to modernize data collection, integration, and sharing to increase the value and impact of the UDS data set and to advance Alzheimer's research. As you know, we are in the midst of a UDS content update. The last one happened in 2015, and a lot has changed since then. These UDS updates are critical for advancing the field, and we're honored to have the opportunity to collaborate closely with the clinical task force on this effort. It's important that the UDS content be updated to reflect new research and technology developments in the field and our evolving understanding of best clinical practices. All of this work is happening under the leadership of and in close collaboration with the clinical task force led by Alan Levy. We collaborate on meeting agendas, setting strategic goals, and tracking deliverables. This time around, the UDS update is more than just a content update. It's going to be different because it's going to be accompanied by a lot of other big changes at NACC, all of which are aimed at modernizing our systems and better serving the ADRC community. Today, I'm going to give you a preview of some of the modern UDS-related tools and resources that we're developing in collaboration with others in the community, including a new electronic submission system, a new pipeline and database that will be accompanied by additional data integrations, as well as a new search and access interface. The existing submission system at NACC is 20 years old and in desperate need of an update. We want to make it easier for ADRCs to submit data. This is going to involve a modern submission system, putting a modern submission system in place that is cloud-friendly and virtualized. In addition to enabling electronic data capture, NACC will continue to host regular batch submissions for the sites for UDS v4. We're starting with REDCap because this is the canonical tool for forms data capture. In addition, 29 centers have indicated that they're either already using REDCap for this or are very interested in using it for UDS v4. In the future, all UDS data will go through REDCap at NACC through one of these 3 mechanisms: direct data entry into NACC REDCap, synchronization of local ADRC REDCap instances through REDCap APIs, bulk upload of CSV files into NACC REDCap. NACC is eager to co-develop an electronic data capture system in collaboration with the community, the ADRC community, to ensure that it effectively serves the community's needs. We do not want to build it and hope that you come. We are collaborating closely with the ADRC community to put an effective electronic data capture solution in place. NACC, together with the Data Core Steering Committee, launched an EDC work group last January. We are currently collaborating with 45 people across 17 ADRCs to co-develop REDCap forms for UDS, define requirements, and to develop training and education materials. I want to thank all of the REDCap workgroup members who have been contributing to this effort. It's been a huge effort, and it's just getting going, including my co-lead, Sudeshna Das, as well as the co-leads for each of our subgroups, which includes Dr. Carrie Stevens, Dr. Ben Keller, and Laura McLeod from the NACC team. So a lot is changing with the Centers program, and we also need a modern database to support these changes that is aligned with NIH's overall data science strategy. NACC is actively working to build a new pipeline and database that will not only accommodate UDSV4, but also effectively scale to collect and integrate new data streams over the course of our current award cycle and provide a robust, long-term resource for the community. Dr. Sean Mooney and Dr. Ben Keller at NACC will be critical in putting this in place along with their teams. So in the past, NACC has collected these data streams from the center. So longitudinal clinical data, neuropathologic data, legacy, MRI, and PET data, and then connected it to NACC IDs. In the future, we will be directly collecting at NACC digital biomarker data, digital neuropathology data, as well as EHR and CMS data. We also want to expand the amount of metadata that we integrate from SCAN, NCRAD, and NIGADs, and connect this data to NACC IDs for all ADRC participants. Our ultimate goal is to make all of this available through our data front door, which will serve as a one-stop shop for all ADRC data and be open for any investigator around the world to use, with everything connected to NACC IDs. Once again, we don't want to build it and hope that you come. We want to build a resource that is going to be responsive to the community's needs and will advance the field. We have recently launched a requirements pilot project with NCRAD and NIGADs, and we're eager for input on your needs. I want to go through a couple of different ways that you can provide input on this now. So you can visit our interactive data front door station here at the conference. Several of us will be over, including myself, in the Solana room from noon to 1:30 today. If you see a bunch of big sticky notes on the wall, that's why. That's our interactive station. We're also encouraging the community to sign up for one of our focus groups that we're going to be doing in the coming months in collaboration with NCRAD and NIGAD to better assess needs, what people want for this data front door. In addition, you can scan this survey right now to provide input. And that's all set up and ready to go. So ultimately, we hope that this data front door will be easy to use, self-service, cohort discovery enabled, real-time, and integrated. So here's how it all fits together. There's a lot going on. We're collaborating with the clinical task force on this updated UDS content. We're collaborating with our EDC work group to set up a streamlined submission system. We're going to be working with a future collaborator to develop this integrated pipeline and database. And we're collaborating with NCRAD and NIGADs, and hopefully all of you, to develop an expanded search and access portal that will connect all of these different data streams to NACC IDs. Having all these different data streams connected by NACC IDs and searchable through a single data front door is going to hopefully enable researchers to ask new big questions in Alzheimer's disease and provide a large set of integrated and heterogeneous data to be able to answer them. So when will all this be available? So there are some timing constraints here. We are working with the CTF on the content update, and we're still designing and building out some of the architecture for some of these components that I mentioned to you. But we are 100% committed to keeping you in the loop. We will be providing regular progress updates to the ADRC community. All of you should have received an email update on UDS last February. We will be continuing to send those. We have set up very transparent tracking on the progress of all of this through our shared CTF tracker and forms tracker. We will be sending updates through email, through our new newsletter, as well as on our website. And we're committed to providing early forms access for testing with your systems. But we also want to know, how do you want to receive updates? So yet another QR code here. You can scan this, give us some input on what would be the most effective way for you to receive updates as we proceed with this work. You can also join our webinar on June 3rd, and to learn more about the status of the forms, another QR code, you can go ahead and sign up there. I will mention that all of these QR codes are also listed in your packets if you didn't get a chance to snag it while it was up on screen. So thank you, everyone. I want to thank all of you for being a part of this, as well as want to thank the NACC team, and of course, my boss, Bud Kukull, for helping to enable so much of this. Okay. I have one last thing to say, sorry, before. So we want to keep iterating on this event and making it better and better. We can't do that without your input. So we have worked very hard, the NACC team has worked very hard to put together some surveys to collect your input on each of the sessions, as well as on ways for you to contribute and share your thoughts on some of the initiatives that I mentioned to you. So please, please, please give us your feedback, share your thoughts. If you go into your folders, as Therese mentioned yesterday, this is available. And you can go ahead and give us your feedback that way. Thanks so much, everyone.
The next speaker is Dr. Jeff Dage from NCRAD.
I'm so sorry not to be with you at the first ADRC directors meeting since we've been coming out of COVID. I'm with my family at my son's graduation, but I couldn't resist the opportunity to be able to share with you updates on what we've been doing at NCRAD during COVID. I want to start by reminding us of our effective partnership that we have together. We have partnered together for almost 2 decades to centralize DNA from ADRC participants at NCRAD. That has allowed us to work together so that NCRAD can provide to the ADRCs APOE genotyping from the samples that we have here. We've been able to partner with the ADGC to obtain GWAS for all individuals that have a sample at NCRAD. We've also been able to partner with the ADSP to have whole exome sequencing and whole genome sequencing on a subset of the ADRC participants. We have been able to work together so well that we expanded our efforts just before COVID to establish the Alzheimer's Disease Center Fluid Biomarker Initiative. This initiative is a blood-based initiative, but expanded beyond just DNA to now bank not just DNA, but RNA, plasma, serum, and PBMCs from the well-characterized ADRC participants. These samples are central to new research discoveries, and as with all samples at NCRAD, any data generated from these samples has to be shared with the research community. This has been an incredibly productive partnership despite the challenges of COVID. We have been able to bank from samples that you have sent DNA and plasma from over 2,000 individuals, and we have PBMCs from over 600 individuals. We've used the rich data available at NACC for all of these individuals because there's a UDS to break out the numbers of samples that we have available by diagnosis. So you can see the richness of the data that we have linked to the samples that are now with us. If you want to be able to look at this in a little more detail, you can look at this either through a NACC, looking at the UDS dataset, or by looking through our NCRAD catalog, where we have a subset of the data at NACC available through our catalog. You can also look at the genetic data that's available on these participants, either broadly through NIAGADS or through a link that we have in the NCRAD catalog that shows you who has data at NIAGADS. I want to thank all of you for your help in our renewal of NCRAD that was funded a little over a year ago. But it's clear that one of the things we need to now prepare for are plasma biomarkers, which are such a key part of ongoing AD research. We believe that we can partner with you effectively because we've been able to establish uniform sample collection and processing protocols. And so because of that, we've established, through the help of NIA, the NCRAD Biomarker Assay Laboratory that allows us to take samples banked at NCRAD and rapidly be able to generate plasma biomarkers for those samples. One of the things that we can also do through this new laboratory at NCRAD is partner with studies that perhaps don't have others with whom they can collaborate to obtain plasma biomarkers, and we can help them fill in that hole. Jeff's going to talk about this in a bit more detail, but we're limiting our efforts to just a core set of plasma biomarker assays that are commercially available and for which we can ensure that we can support longitudinal quality monitoring and have consistent results over time. We started with 4 initial assays for p-tau, NFL, A-beta-4 and A-beta-42, and we're now expanding that with some additional validation. Something that's very important is the ability to be able to combine data generated from multiple laboratories. We saw how important that was when we first started doing GWAS arrays, and it's equally important now that we're focused on plasma assays. We need to be able to combine data at the back end so we can generate large sample sizes and be able to ask key new questions. We want to help partner with that goal and be able to work with different laboratories that are generating these assays to ensure that we have the ability to compare results across laboratories and platforms. We have consistent and uniform reference standards that we're using, that we can standardize and test pre-analytical conditions, and we can ensure quality control and performance testing. Right now, thanks to support from NIA, we were able to obtain several administrative supplements that allowed us to establish the laboratory and purchase the equipment, hire the staff that's now working in the laboratory, and we're in the process of generating these plasma assays for 4,000 ADRC samples. We have 2,000 that we got through the ADCFB that I just showed you, and we have availability to do another 2,000 that can be either samples that are transferred or that we're in the process of transferring from the ADRCs. We'll also be running 2,000 Indianapolis and Baden plasma samples. We're going to be submitting a competitive revision in the fall of 2022, and we're requesting funds that will allow us to continue to support our ability to generate plasma assays going forward over the remaining years of the NCRAD grant. Right now, our administrative supplements are just paying for this to continue until the end of next year. These additional funds will also allow us to support centralized banking and plasma assays for additional studies, and we're particularly focused on those enrolling diverse participants. We have a website if you'd like to learn more about what we're doing in the laboratory, or if you'd like to request a quote or a letter of support to include in a grant. And lastly, I hope that you're very excited about what you've heard and you'd like to learn more. Jeff will be available to answer questions in the next part of our presentation, but I also want you to be looking for Casey Lacey, who's in the audience, and I'm hoping she's raising her hand now. She would be glad to talk with you, explain more about how we're working with studies to bank samples, and in particular, if you'd like to learn more about how we can work with you to help get plasma assays done through the NCRAD Bioassay Laboratory. Thanks so much, and I hope you enjoy the rest of the meeting.
Yeah, so clearly that wasn't Jeff Dage. I'm Jeff Dage. I'm really happy to be here too. This is my first time coming to an ADRC meeting. I just joined Indiana University just last August. Before that, I was at Eli Lilly, for anyone who doesn't know me, working on blood-based biomarkers and translating them into the clinic. And so I'm going to take a few minutes and dig a little deeper into what we're doing in the biomarker assay laboratory within NCRAD and explain a little bit about the purpose. Why are we doing it? And Tatiana touched on that. And the real mission is to provide the reliable and consistent research biomarker results. And so we're focusing on just a few assays, but we're going to do those assays really carefully and really well so that we get reproducible results over time. Because what we need to do in the blood biomarker space is to start using them. Stop characterizing them, stop testing them, but use them to make some important scientific contributions. And I think we can start doing that with some of these biomarkers. And so it's a really exciting time, and it's happening pretty quick. But that's the main purpose. And as Tatiana mentioned, and I mentioned earlier, we're focused on just A-beta 4240, neurofilament light, p-tau. And now we're just in the process of validating GFAP, and we're looking at the singleplex or the duplex or the fourplex Quanterix assays. And so, as Tatiana also mentioned, we want to keep the end in mind. And there's a lot of issues that have ruined blood-based biomarkers or even biofluid biomarkers for a long time, is we get bogged down into issues of pre-analytical conditions, which tube types, stability, collection, fasting, not fasting. You know, how long does it take you to centrifuge it, and how long do you centrifuge at room temperature, 4 degrees? There's all kinds of little issues that raise confusion, and those kind of variability can prevent us from ever moving forward. And that's really what we're going to be trying to accomplish, is to implement these, do all those tests, make sure that we understand it, make sure we can inform you what the real issues are, and where we have more flexibility. And what we call that is assay robustness, right? How robust are these assays to different variables? And so we'll be doing that within the NCRAD biomarker assay lab. And again, the only reason we're doing it is so you can use the tests, right? And you can trust the data. We have already developed a cut point, and we're looking at the stability of that cut point, and you'll be able to use that cut point. And so we'll have it published. You'll be able to pre-specify your cut points and how you use it. Those kinds of things will add value to your studies, and that's what we're trying to accomplish. And so in order for us to do it, as there's this proliferation of assays, platforms, and I'm going to talk about a lot of these at the biomarker core meeting, you know, there's a lot of laboratories that are running them. And so just like we've seen with imaging and scan and clarity, as Tatiana mentioned, genetics, we need to standardize, we need to harmonize some of these measures. It doesn't mean everyone has to do the same thing. We have ways of bridging data together. We have ways of controlling. We want to implement a quality assurance program where we test, or people who want to be tested. We can send samples blinded to different laboratories who want to be part of it and see how their performance tracks, so that in the end, when all the data does go to NACC, we can merge it together, we can do these large analyses, we can use it as an endophenotype for our GWAS, or we can combine it with all the data from SCAN and Clarity to do some really impressive analyses. And I think that's only going to be accomplished if we can really harmonize and standardize and get people who are generating large volumes of data on board and into that program. And the program doesn't exist. We're just in the middle of it. So if you're interested, definitely reach out, because we would like to kind of make it, you know, as many centers who are measuring these things that want to be part of it, we want to include you. The intention is not to exclude anybody. And I really believe that this is going to advance biomarker innovation. And so if we can lock down, if you consider harmonization locking down, start using the biomarkers, it's going to enable us to find novel synaptic biomarkers, novel markers of neuroinflammation, and other things. And really that'll help drive us to that next generation. And I think that's my last. Yep. So there we have questions. As Tatiana already mentioned, there's people here. I'm happy to answer questions too. Yep. Oh, the biomarker core meeting is later today? Oh, come. Yes, definitely come. So I'm going to be giving an overview of blood-based biomarkers. So if you're interested in kind of my view of the status, I'll be doing that.
Yeah, Charlie at UC Davis.
I must have went over time, or Charlie doesn't get to ask a question. I don't know. Oh, okay.
Tatiana knows what I'm going to say. And I think that it's something that I've struggled with
the NCRAD, okay? So just give you an example, we have over 500 RNA samples in very well-characterized individuals from our ADRC. We have neurofilament light. We have those all on our active cohort. And yet you all don't, we have DNA as well, that was done outside of the ADSP. And there's no method for you all to take advantage of that. And I think that I'm not alone. I suspect if I look around, and I see other people nodding, I'm not going to send you those NCRAD. I'm not going to send those samples to NCRAD, right? But I'm willing to, share and let people know. And I just wonder how many people here feel the same way, and how, you're talking, and this is something that we're really moving toward. We're talking about doing this, but you're demanding that we give you the blood so that you do it. And I just find that a little bit unsatisfying. And I don't mean to accuse you, but that, okay, Cerise is going to come beat me up.
No,
here you go. No. Beat me up, Cerise.
Well, here, I can answer that from the biomarker lab perspective. For sure, what I'm talking about, standardization, harmonization, it's about creating a federated set of laboratories. And so you don't have to send the samples here, and Tatiana mentioned that as well, is you can run them in your own lab. But all I ask, and what I'm asking, is that, participate in raising the level of validation so everybody understands the kind of precision you get over time in your lab, raise the level of quality so that when we merge the data together, we can do that and we can include all of the results. And it's about the data more than the samples from the biomarker perspective. Now, there's not enough capacity in one lab to run all the biomarker samples that need to be run.
Jeff answered it very nicely, so I won't continue. Anyone else have a question?
So which phospho-tau epitope are you using, and are you planning to do more than one?
Yeah, so I'm the inventor of the phospho-tau 217, so that would be my choice if it was commercially available. And what we're trying to do is implement assays that are available for the long term, right? Because we want to implement these things over the years. Right now, the only commercially available assay really is the Quanterix p-tau 181 assay that was developed in Sweden by Kai and Henrik. And that's what we're implementing now. But there's a series of new assays, 231, 217, coming from multiple places. And even ADX Neurosciences has a 231 and a 181 assay. Within my research lab, not in the NCRAD biomarker lab, I'm evaluating those, and we'll be testing those on the same samples that we use for validation. And when we see one that can perform and perform well, and it becomes available commercially so that we have years of, longitudinal data that we can look at, not that we can look at, but that we can anticipate, we'll be able to implement those, and that'll be the plan. So we'll always be looking for the best assay that's commercially available. And then when we do change, because we will change, we will do the bridging necessary, we'll do the harmonization, and we'll do the alignment of cut points so that you know when we've switched and how that all comes together, so you can still bring the data together in the end.
Hi, Howie Rosen from UCSF. There's been an ongoing discussion that we've had here about disclosure, and so I'm wondering. Rob reminded me it's very complicated, but I wonder if there's been a lot of thinking about moving toward CLIA certification so that at least we'll have that potential ability to disclose.
Yeah, absolutely. I think that's a great point, and I know that was discussed yesterday, and there's several work groups looking at that. And I know of, there's appropriate use criteria being drafted already. So, there is a lot of interest in that. What we're implementing in the biomarker lab is research use. I mentioned we're partnering with C2N to enable studies who want to do reporting to have that. We'll be doing the same thing with our phospho-tau measures, identifying CLIA-certified laboratories to run those. So when that is something of interest, we can do that. The research lab, we probably won't pursue CLIA. We'll do that through partnerships, but we'll, again, harmonize, bridge. So if you get data from our lab, you get the data from that CLIA lab. When you merge it together from a research perspective, you'll be able to do that. It's a great point.
I had the same question, so.
It's a good question. Thanks.
We do have a few questions in the chat that I'd like to pose real quickly. One from Anika Norin. Is the NACC ID the PT ID or something new NACC is proposing? Maybe we can give the microphone to Dr.
Jeff.
Biber as well.
Get Sean. Sean? Where's Sean?
Would anyone like to answer that?
Can you repeat the question?
Sure, and maybe Dr. Mooney can answer that as well. Is the NACC ID the PT ID or something new NACC is proposing? Yes, is the answer to that.
Please, I can answer the question.
Sorry, hiding on the floor. The answer is neither. The NACC IDs have been in use for a while. They're the common identifier for the aggregated data. Once your data comes in to NACC, we assign the NACC IDs, and that's how we identify the participants.
Okay, do you have any other question?
One more from Boris DeCourt. Will there be a possibility to use the cloud version of REDCap to have all the fields pre-set up and the database of each ADRC automatically transferred to NACC?
This is a great idea. Send me an email. I'll connect Boris to the REDCap team, and we can talk about doing it. But it's an awesome idea.
Okay, thank you. Before we head to our break, I have two very important announcements. I will be announcing the awardees for the Rising Star Award and for the NACC New Investigators Award. Thank you for all the 2022 Spring ADRC nominated presenters. We appreciate you taking the time to share your work with us. We would like to recognize the top 2 presentations as voted by the attendees of the spring meeting. Each of the awardees will receive a $1,000 stipend provided by NACC. And the awardees are, do we have the slide? I'm just waiting for the slide. Okay, it's Nicole Ragus-Puglia from Wisconsin ADRC and Miranda Lim from Oregon ADRC. I also have the pleasure of announcing the winners of the New Investigator Award for 2022. The New Investigator Award program was developed to support new and early career investigators as they further their research careers in Alzheimer's disease and related dementias. This year, the NACC received 51 applications from 29 different ADRCs, the most ever for the New Investigators Award program. This is huge. We would like to thank to the many reviewers who contributed their time and expertise in reviewing the large number of applications. The steering committee, at the steering committee meeting, we selected the winners of this year's award, and we are excited to announce the 3 New Investigators Award. Each New Investigator Award will receive $135,000 to support their outstanding research projects. And the awardees are Andrew Young from USC, Yukiro Katsumata from Kentucky, and Jaime Ramos Cejudo from NYU. Well, now comes the break, and we can reconvene at 10:15. Oh, Sarah, come before we leave. Then we go to the break.
Sorry. So I just had one more, small thing to say, which is that we thank the entire NACC team for all of the incredible work that has been put into this conference, making this possible. But I wanted to give some specific shout outs to the operation team at NACC that has been absolutely instrumental in working for weeks and weeks to pull this together. So I wanted to give some recognition to Hannah Rosenstrader and to Amy Young. Hannah's unfortunately not able to be with us today, but Amy should be here somewhere in the room. There's Amy. Thank you. I also wanted to thank Dr. Melissa Trapp-Petty, who is new to the operations team and has been phenomenal. Where is she? She's not here yet, but okay, we'll thank her later. Thank you, Melissa. Is she here? Oh, there she is. Yay! And then also Brittany Hale, who's also very new to the team. She's virtual right now, but she's behind the scenes. You'll see her all over Hublot. And then also Claire Cassidy, who I believe is here somewhere. Okay, well, we can all thank Claire later. Oh, there she is. Yay, thank you, Claire. As well as Val Hawkins, who has been behind the scenes helping to pay for all the things. So, is she here? Okay, now we can go on the break, but I just wanted to make sure we did those shout outs. Thanks, everyone.
Hello. I think we can start. Hello, we are going to start. I'm going to introduce Kate Possin from UCSF. She's Associate Professor of Neurology, and is going to give us an update or summary of the ADRD Summit.
Thank you, Oscar. It's a real pleasure to be here. I know many of you were involved in the ADRD Summit, either as committee members or providing your input in other ways. Thank you for that input. For those of you who have not been involved, I hope I can encourage you to get involved in the future, because this is how we can use your scientific expertise to shape our research priorities. So there are 3 summits that are led by NIA and NINDS. Those are the AD, ADRD, and CARE summits. Each is held every 3 years. And these think tank summits result in research recommendations that are turned into milestones after NIH Council approves them. These milestones then guide funding opportunities and our scientific advancements towards Goal 1 of NAPA plan to prevent and effectively treat AD-ADRD by 2025. We have to work hard. So while NIA leads the NIH response to the national plan and leads the AD and CARE summits, NINDS leads the ADRD summit and also the portfolios on LBD, FTD, VCID, and MED. Yet NINDS and NIA collaborate closely on the funding opportunities, on the supplement program that you heard about from Richard Hodes this morning, on the pay lines, and on these triennial summits. So this year, the ADRD Summit was held in March as a virtual summit for the first time with our scientific chair, Natalia Rost, and our NIH lead, Rod Corriveau. Keith Whitaker, who's here, was also central to driving all of this work. There were a set of topics you can see on the slide here, and there was a committee associated with each one of these topics to generate the research recommendations with substantial input from a variety of stakeholders. So the summit topics were health equity, FTD, VCID, LBD, MED, and also we had 3 special topics as well. Each committee worked together over a 6-month period with broad stakeholder input to draft milestones that were presented at the summit for open forum discussion and further public input. These have now been finalized by the scientific committee. They are now in the hands of NIH. They will be approved by council and turned into milestones to inform the AD bypass budgets that are delivered to NIH by Congress. It was by far the most well-attended ADRD summit. I think because it was a virtual meeting, it was more accessible to more stakeholders. Whereas prior summits had less than 500 attendees each, this one had over 1,500, with substantial engagement that really impacted how these recommendations have been finalized. Some common themes across the summit, across all of those different topic sessions: the importance of achieving health equity, the need for implementation of pragmatic approaches and solutions, increased focus on pragmatic clinical trials, the need for precise biomarkers to identifying underlying disease processes across all ADRD, personalized approaches to prevention and treatment with health equity as a central focus. In clinical research, concerted attention should be paid to the immediate needs of individuals living with these diseases. And in basic research, the needs include novel strategies and tools and seeking synergies. I'm now going to give you a little bit of a window into one of the topic areas, multiple etiology dementias, or MED. So what is, before I tell you what MED is, I just want to comment on the incredible group of scientists that were part of this committee, you can see listed here, as well as the NIH program staff. Our lead was Linda McGavern, who worked tirelessly together over 6 months to produce these recommendations. So now, what is MED? Well, in dementia, mixed underlying etiologies is the norm. Over 50% of people diagnosed with AD who were studied at AD research centers have pathologic evidence of more than one cause of dementia. The likelihood of mixed dementia is even higher in community settings and in the oldest old. If we are going to accurately diagnose and treat Alzheimer's and related disorders, we need to understand these contributing pathologies, including how they can be identified, treated, and even prevented. And we must continue to advance research on differential diagnosis, treatment for people with multiple etiology dementias, basic science on the common and interacting disease mechanisms, and training in these different dementia syndromes. In addition, the AD-ADRD field, some critical research gaps apply across all etiologies. These are also subsumed in the MED session. These include early detection of cognitive impairment, for example, in primary care, caregiver support, and data harmonization. So in summary, what is MED? MED recommends research on mixed dementias and cross-cutting themes. So I'll now introduce you to the focus areas of the MED topic area, starting with detect and diagnose. More than 50% of people living with dementia are not diagnosed until moderate or advanced stages. These delays are longest for race and ethnic minorities. We need scalable solutions to achieve diagnosis that are timely, accurate, compassionate, and actionable. Interventions and treatment. We recommend research that moves forward promising interventions and treatments that address modifiable risk factors and that support people living with dementia and their families. Basic science. MED is the norm among older adults with dementia. Although much of basic research focuses on a single etiology, there have been substantial advancements in understanding the interacting and common mechanisms of mixed pathologies. Continued work in this area is crucial if we're going to develop accurate methods to diagnose and treat these diseases. The MED group also recommends work to address gaps in the dementia clinical and research workforce. And harmonization. This is new to the ADRD Summit this year. We recommend research to improve data harmonization and sharing practices to maximize the impact of the data we collect. Infused through all 5 of these focus areas is the requirement that research be inclusive. Research should be conducted in a way that is culturally sensitive and relevant to the populations who suffer from MED. So each one of those topic areas I mentioned, MED being one, the others being LBD, for example, or VCID, each one came up with 8 top-line recommendations. And we had to rank those in order of priority, which was no easy job. I'm going to share with you now the 2 top priority recommendations for MED. Number 1 is to evaluate pragmatic approaches to objectively detect cognitive impairment and link to quality care. So this recommendation was first emerged, a version of it, in the 2016 ADRD Summit. This led to funding a consortium of projects called Detect CID that is focused on improving detection of cognitive impairment in primary care. There will be a new round of these awards funded, I think, later this year. It'll be even a larger consortium, I hope, by the end of this year. And this is a perfect example of how research recommendations from these summits are translated into funding opportunities and advance the science. The other top priority recommendation is to conduct clinical studies on approved or promising interventions and treatments to mitigate risk for cognitive decline. It's estimated in this seminal paper by the Lancet Commission that the 12 most modifiable risk factors account for 40% of worldwide dementia. So we need to leverage this research to develop and test interventions to help prevent or delay cognitive decline. So in closing, here's where you can learn more. Please go and check out the ADRD Summit recommendations to see where the field is going. In the second bullet there, you can see the upcoming funding opportunities with NINDS, much of which are really driven by summit milestones. And lastly, the ADRD pay line. The take-home point is if your grant is funded at a 28th percentile, you have a good chance of getting that money and moving forward with your work. So thank you. I hope I've convinced you all of the importance of these summits for driving research priorities and funding for our field.
Thank you, Kate. Very informative, very nice. So we are going to start now with a session to honor John Trojanowski, and Dave Walk will be the first speaker.
Hi, everyone. It's really an honor to be able to speak about John today, and I really want to thank Oscar and the organizers for putting this together. I think it means a lot to many of us here. He meant so much to the people at Penn and certainly to the ADRC program. He actually was the inaugural director of the Penn ADCC in 1991 and served that role for 30 years. His reach obviously goes beyond this room, though. He has done really foundational work and fundamental work in PD, ALS, and FTD, as well as Alzheimer's disease. And as Nina mentioned earlier, he was really a champion of bringing together those different disease states and thinking about how they interact. And as we just saw, most of dementia is multi-etiology. You're going to hear a lot about his research accomplishments today, which were obviously prodigious, and he was incredibly productive, really from the beginning to the end of his career, up until the last few years of his career. But what I wanted to do is just provide some of my own personal reflections on John, who definitely in the last few years had become a close friend. I should start by saying that it's not really lost on me how irreplaceable John is as the director of the Alzheimer's Disease Research Center at Penn. He was an incredible leader, and it's really a daunting task to follow in his footsteps. In fact, Eddie Lee and I joked when we also took over the Penn Institute on Aging with John that it really takes two of us to equal maybe like the half of John's energy, or maybe even less than that, because he was such an energetic person. Many will say he was a giant or a towering figure in the field, and as many of you know, this was more than metaphor given his height. I'll never forget when I returned to Penn to interview for a faculty position with John. I was most nervous about meeting him. I didn't really know him that well, even though I had done residency at Penn. And I'll never forget that he sat in a stool, and I was in a chair. And so why, a 6'4" person would decide to be in this higher up stool while I was in, somewhat more diminutive person was in this chair. It just added to this intimidation now as I was looking up to this literal giant. But that sort of asymmetry, I think, just quickly faded away because he was just so kind and really generous with his interest in me and my plans and my research. And to some extent, I think that was John's sort of special sauce, is that he really had an innate curiosity about everything. And he could talk to you about any topic and would be interested in any topic. I think, he described himself as a people person, and I think he really was, and that's what made him such a champion, I think, of team science. Succession planning was never an easy topic for John. As an example, he confided in me when he won the Lifetime Achievement Award from the AIC that that he hoped people didn't think that was the end of his work, because he was going to continue to carry on. He said it multiple times. But I have to say, when we decided to move on to our succession plan at Penn, he couldn't have been more kind and invested in that transition. He went over the grant with me multiple times. He gave advice not only on the center, but on career and leadership. Really was just incredibly engaged. And actually, I think probably more than any of us was the most happy when we did well in review. It was personally gratifying to me to see, this father figure as such a proud parent of what we had accomplished. A few things about John is, we all have little quirks or foibles, I think, that people characterize us by. John had, maybe more of them than others, I think, in terms of these quirks. As Eddie pointed out in his beautiful piece in Acta Neuropathologica, John was really, as you can see in these pictures, a stylish, sort of debonair guy. He's famous for his colorless shirts and his, luscious hair, which as I, started to lose mine, I became a little resentful of that over time, but, I can get over that. But despite the fact that, he had that style, he also wasn't afraid to wear a fanny pack almost everywhere he went. And so it was an interesting mixture. He was known for that fanny pack. Another thing, that I think all of us realize here is John was always the first person to ask a question. These slides are advancing without me. Well, but he was always the first person to ask a question at any talk. The talk could be about paper mache, and he'd ask probing questions about paper mache and how it was, evolved over time. I mean, he just was, again, so interested in anything that he could try to learn or listen to. And of course, he would always introduce himself, even in a room where everyone knew him, as John Trojanowski from University of Pennsylvania. And we're like, we know who you are, John. He, likewise, for those of us who worked with him closely, and probably many in this room, he also would send you an email that was basically like multiple paragraphs long, often, each paragraph a sentence, because he had a tendency to write these run-on sentences with 5 articles attached to it. And then inevitably, in like 10 or 15 minutes later, he'd forward that email to you again, have one sentence at the top and that he added to it. Then another 10 minutes later, he'd send another one with more articles, and that would go late into the night, and you'd wake up the next morning with these lists of articles and emails from him. He did the same with phone messages. He left phone messages that, if you took the median, was somewhere north of 2 minutes. If you took the mean, it would be like, much higher than that, because there were definitely some outliers on those calls. And usually it was him just calling about some idea that he was excited about or something he wanted to chat about. And there, too, he would always say, "Hi, Dave, this is John Trojanowski calling." And, I was always like, okay, you could have just said John. I know I recognize the voice. And you'd usually end with, no need to, get back to me right away. We can talk about this later. And then inevitably there'd be another phone call 10 minutes later with another minute or two message sort of adding to what he had just previously said. And again saying, "No, this is John Trojanowski again, giving you a call." The other thing I think many of us, and Charlie and I were talking about this last night, that he's famous for with his wife, Virginia, is they occasionally would disagree about things in public. They'd have they'd bicker. The first few times that happened, with me, I was just like, what's going on here? But it was always extremely short-lived, and I have to say they always left together happy, and it kind of made you feel like you were part of the family, because they were unfiltered in that way. Really, when I think of John, one of the things I think about is just the word joy. He had incredible joy for his work. He loved it. He loved talking about it. He loved every aspect of it. I think he got a lot of joy from his colleagues and friends. He was very proud of them. And then most of all, he had incredible joy around Virginia. At his celebration, he had a celebration shortly before he passed away of his accomplishments. He obviously thanked his life partner, Virginia, and described her as, or said that they were not only kind of joined at the hip, they were joined at the brain. And that, I think, is really true. And actually, kind of beyond their accomplishments, they really just genuinely loved to be together. They just liked to talk to each other and enjoyed each other's company immensely. And it really is, quite inspiring. I mean, we should all be so lucky to have someone that we just really enjoy spending all of our time with. And they certainly did that. It's really hard to think of coming to these meetings and not seeing John. As we do more and more events where he's always sort of in my mind, it becomes hard to kind of fathom a world where John is not a part of it. And I'm sure many of you share those same feelings, waiting for him to ask the first question. But I can say that this community and beyond has been made really a better place because of John, and I think we've all been touched by him, and we'll really deeply miss him, and we'll continue to miss him. Thank you.
Hello, Dave. I'll get your mask back later. So my name is Eddie Lee. I'm a neuropathologist and scientist from the University of Pennsylvania. I met John over 25 years ago, and I've been working with him since, until his passing earlier this year. So I've been asked to talk about his insights and impact from a scientific perspective, and so I'm going to give you a broad overview of what he's done over the last 40 years, and there's a lot. And I kind of divided this into four subjects. The first is that he really championed neuropathology, and what I mean by that is he showed us that studying the human brain, autopsy studies, studying the tissue, can actually give us understanding about the disease. It could drive disease. And to give you some perspective, this has actually been a debate in academic American medicine for over a century. So in the turn of the late 1800s to the 1900s, there was actually a debate about whether pathology was valuable in terms of understanding science. And people actually thought that the clinical pathologic correlation was going to stop being useful by 1900. And these sentiments that pathology is observational and not mechanistic are still pervasive today. But John, despite this, thought that it was important to study the human brain and to do pathology. And so this is actually one of the first brains that John ever banked in the brain bank. And this is in the 1980s, talking about somebody with Alzheimer's disease. It's almost comical thinking about the workup they did with about 5 sections and 3 stains, where he was able to see plaques and tangles, and he also actually saw some Lewy bodies. And I'll come back to that in a little bit later, but called it senile dementia of Alzheimer's type. So to give everybody a sense of the criteria that was used, this is our current criteria, the NIA Alzheimer's Association guidelines for the neuropathologic diagnosis of Alzheimer's disease. And we have to wind back to the NIA Reagan, to NCRAD, to back in the Cacciatarian criteria days. And so this is the era that John began banking brains at the University of Pennsylvania. And so he became faculty in 1981, and so this is his first faculty picture. In the background there, you see other greats, Nick Ganatis, Len Jarrett. Peter Knowles in there, he discovered the Philadelphia chromosome, and he said cancer is caused by genetics. And so it's a heady group of people, and if you can't find John, he's up here in the corner with this nice bow tie and the mustache. I did not see him at that time. And so in terms of his scientific accomplishments, I'll fast forward a little bit to 1991. This is a review article by Dennis Salko commenting on the state of essentially biochemistry and pathology and Alzheimer's disease. And in that review, he said, since the quantitative depolymerization of PHFs have not been reported, we can't be sure if tau was the only protein in neurofibrillary tangles. And if you look at the date, this was published in April 1991, and publishing back then was a lot more difficult than now, right? So if you have to make edits, after a certain point, you can't do that, right? And so it's actually very interesting that this came out in Neuron in April 1991, and just a few months earlier, Virginia and John had actually done what Dennis Salko said was missing. And so in this paper in Science, using the brains from John's brain bank, they were able to purify paired helical filaments to homogeneity. And so on panel B and C, you could see the electron micrograph showing these beautiful paired helical filaments. In panel A, you could see a protein blot of the different fractions. And in the third lane there, you could see that they purified tau. And for the biochemists out there, this is not an immunoblot. This is a Kumasi stained gel, right? So this is total protein where they purified only tau in these filaments. And so they kind of put the nail in the coffin saying that tau is the building block of paired helical filaments. And so, of course, a lot of the work was done with Virginia, his lifelong partner. But one thing I do want to highlight is how he did work with many of the scientists in our field today. So if you fast forward a few years, Maria Spillantini was a scientist in the UK. She just started her own independent laboratory. And at that point, they just discovered that alpha-synuclein mutations cause Parkinson's disease. So Dr. Spillantini had some antibodies against alpha-synuclein and started staining tissues and thought that Lewy bodies might be made of alpha-synuclein, but she didn't have enough of the tissues. She didn't have dementia with Lewy bodies and other things. And so she reached out to John, and the story goes within days, I think, or MTAs really weren't a thing then. He sent over cases, and for the first time, we saw Lewy bodies are made of alpha-synuclein, and so that was published in Nature in 1997. And then fast forward a couple years later, again with Virginia Lee, discovered that TDP43 protein makes inclusions in the setting of frontotemporal dementia. And this was done with Manuela Newman, who was visiting the laboratory at the time, who we all know as an amazing neuropathologist now in her own right. And so this showed that the missing protein that many people were looking for was TDP43. They found it in a setting of frontotemporal dementia. They saw it in amyotrophic lateral sclerosis. And now, of course, we see it in a lot of settings, including late or in aging brains. And so together, those three proteins constitute the vast majority of the protein aggregates that are thought to cause neurodegenerative disease in aging brains. And so to harken back to this champion of neuropathology ideas, I'll put this quote here that was published in the Annals of Neurology in 2007 when John of Virginia discovered TDP43. And so you could see the same argument saying, hey, does pathology matter? Is this just junk? Where's the mechanism here? We're still being made now. I sit on study section and you could hear it all the time. But despite that, John stuck to his guns. And if we go back, for example, in this original autopsy report that he saw, we've actually revisited all of our cases, and we know now that the dementia in this case was caused by Alzheimer's disease, but also Lewy body disease, and also TDP43 proteinopathy. So this person had late as well. And now this concept of multi-etiology dementia is commonplace, but that wasn't always the case. And as evidence of this, this is an interactome graph that my colleague Corey McMillan put together. These are all the publications of TDP43 since it was discovered in 2006 from almost 11,000, or well over 11,000 authors. And so each dot here represents an author, and the yellow dot is John. It could just show the impact he's had on this field. And John, before he passed away, had over 1,400 publications, over 5,000 cofactors. His H-index is better than Tony Fauci, and one of the top 10 most cited neuroscientists. So the second theme I had is his contributions in defining what is Alzheimer's disease. And again, we'll harken back to this idea that dementia is complex in the human brain, and it's often multi-etiology. And so if we go back to the criteria, John, together with Brad Hyman, chaired the NIA-Reagan criteria for Alzheimer's disease. And so in that progression from Khachaturian to NCRAD to this one, before this criteria existed, neuropathologists actually did not use tau in the diagnosis of neurodegenerative disease, right? And so at that point, it was very amyloid-centric, and John, Brad Hyman, and many others said, hey, we have to pay attention to tau as well. And if we fast forward, this is a study done with John Robinson, looking at our brain bank, looking at multi-etiology dementia, and of course we see it in the setting of Alzheimer's disease, also in the setting of Lewy body disease and primary frontotemporal dementia and other tauopathies. And I put this here to make the point that this was a common theme in his life about these comorbid neurodegenerative diseases. I also want to highlight that he worked with anybody. So John Robinson is a staff member he's been working with for over 20 years in our brain bank. And so be able to do science with anybody. You're a staff member, you're a junior scientist starting off your lab, you're an established scientist. John was able to make that space for everybody. And then other things that maybe people might not realize as much that John had an impact on our field is how we diagnose Alzheimer's disease. And so he was a biomarker core lead in ADNI with Mike Weiner, and he was biomarker core with Les Shaw. And I just highlight one example where he influenced the field by saying, hey, it'd be great if we validate our biofluid markers based on actual autopsy-proven cases. And so this was an influential paper showing that cerebrospinal fluid, amyloid, and tau can be used to help diagnose Alzheimer's disease. Another example is this study with Dan Skowronski. So Dan Skowronski was a graduate student. I actually overlapped with him. And this was the last paper that Dan published with John of Virginia as a graduate student, where they showed that if you inject a mouse with an amyloid binding compound, it could travel to the brain and label amyloid plaques. And so we know now that Dan was able to start a company based on this called Adrd Radiopharmaceuticals. This led to the first amyloid PET imaging agent. Dan now is Chief Scientific Officer at Lilly. And finally, what dictates disease progression? So can we get into mechanism, how disease progresses? And so this is a picture with John. He had this famous way of showing protein aggregation where he would take a piece of paper and crumble it and kind of throw it down and say, that's what's going on in our brain, and we have to fix that. So that's that paper you see in his hand, with Kelvin Luck, who's a postdoc in the lab. And they showed this is in the setting of alpha-synuclein, that if you inject a mouse with alpha-synuclein, it could seed aggregates in the brain and spread through the brain in a cell-to-cell manner. So this idea of disease propagation has been the driving force for John for the last decade or so. And they've shown this phenomenon for alpha-synuclein, tau, and TDP43 in various experimental models. So that's a quick overview of the scientific highlights of John's career, essentially showing that tangles are made of tau, Lewy bodies are made of alpha-synuclein, and TDP aggregates are made of TDP, but had profound influences in biomarkers, diagnosis, and also the mechanisms of disease. But the title of my talk is Insight and Impact, and I think we could agree on his scientific insights and impact. Perhaps what is a little bit more subtle is that John's real insight is that science is a human endeavor, that he was able to create an environment, a space for him to work with people to allow them to flourish and make their discoveries. And so today we recognize and celebrate John, but also all the people that he impacted, and celebrate their achievements as well. And so this is just a smattering of the people currently at Penn that John directly worked with, had grants with, supported over the years. And I apologize because I actually ran out of room. So there are more people that I would have wanted to include. Of course, there are many people outside of Penn as well that we could add to this montage. Thank you.
Thank you, Dave. Thank you, Eddie. Our next speaker is Pete Nelson. I don't know, he's virtual? Yeah, he's virtual.
Yes, I am. Thank you, Dr. Lopez. I appreciate it. And thanks very kindly, Dr. Lopez. Thanks to the meeting organizers. Thanks to Dr. Wolk and Dr. Lee for their wonderful talks. There'll be a little bit of redundancy, but not too much here. So I'll get right to it. When trying to think of a tribute to Dr. Trojanowski, who was one of my mentors, I reached out to Dino Getty, and he sent along a poem that I thought was evocative. And part of it said, a man, of course this could be a woman, but in this case we're talking about a man, Dr. Trojanowski. He lives for as long as we carry him inside us, for as long as we carry the harvest of his dreams, for as long as we ourselves live holding memories in common, a man lives. And it's in that spirit that I convey to you some of the harvest of Dr. Trojanowski's great works and my interaction with him. I met Dr. John and Dr. Virginia back in the mid-90s. I still remember a wonderful meal that I and my wife had with him and Virginia, and I was just blown away by their glamorous rock star qualities, as I never stopped being. But the 1990s was also a time of change in the dementia work, and John had a big part to play in that. To think about that, I sort of have an analogy that Alzheimer's disease pathology was for a long time dichotomized as no, yes, either you have it or you don't. And similarly, the clinical features of dementia were dichotomized. And then everybody was presumed to follow along either the no, no, or yes, yes portions of that square. It is now recognized that this is not only a construct that is faulty and is wrong and is incomplete and does not capture the complexity of the dementia illness and symptoms and pathologies, but it actually has been holding us back. And this paradigm has actually been a part of why we've failed as a field to get clinical translational success. John Trojanowski has been a big part of the progress in understanding the disease phenotypes, and two recurrent themes that relate to the talk that I'm giving today and the context today are the roles of Dr. Trojanowski and also the large central roles of the ADRC system in general in fleshing out progress in this area. And this is a picture that David Wolk already showed, but I think it's conducive because it indicates what perspective John came from. He came from looking under the microscope. And when you look under the microscope at actual cases, what you see is irreconcilable, completely not able to be reconciled with that paradigm of dichotomizing everything. And so as early as 2001, John was sort of beating the drum about these fatal attractions of proteins that were comorbidly deposited in brains and how they were coming together and in the same brains in these mixed pathologies. But to really sort of get an understanding of that, I'd like to highlight the work that John did, and Eddie did a wonderful job at describing the long, excellent, incredibly productive pathway of John and Virginia. But I'm going to start with the year that I came to Penn. They recruited me in 1997. I came out and started in 1998. And that was a hell of a year for John. In that year, he achieved what most people could not even aspire to. He achieved at that year the Potamkin Prize for Dementia Research. He was the president of our little guild of neuropathologists. He was part of the NIA Reagan consensus document and that huge synuclein paper that Eddie mentioned. But these two customers were actually at this point just really getting started. And I'm going to show you one component of that, how he helped to form and change the way that we even think of dementias, and in no small part in consensus and group projects that were coming to understand how these pathologies appeared. And I'm going to be focusing on this 2006 and 2019 TDP and late projects. But in addition, there was a 2007 and 9 FTLD consortium, 2008 for MSA and other synucleinopathies. The NIA ADNC consensus was 2012, part in 2013, as was RTAG. The DLB consortium was in 2017. So this is really reframing what dementia is. What is the phenotype from the molecular substrate perspective? And importantly, John was a co-author on all of these and so much more. But it gives an idea that the last 20 years have been incredibly productive time for changing the way we even think of what goes on in the brain of people with dementia. Of course, what they are much known for, in addition to some other things, but this was central, was the TDP-43 proteinopathy. This was first discovered on diseases along which we now know is a spectrum of ALS and FTLD. And this work was first published in 2006. Manuela Newman, now in Germany, was the first author. John was an integral component of it. And Dr. Virginia Lee was the corresponding author of this work. And so this really became sort of the paradigm when people think of TDP-43. And indeed, these diseases are largely related to this pathology. However, it's now known that at least 20 other disorders. This is a promiscuous pathology, and wherever it's present, it is problematic in brain trauma, in various other neurodegenerative conditions, and also in developmental, neoplastic, and other diseases. There are many different TDP-43 pathologies, of which the most common is late, limbic predominant, age-related TDP-43 encephalopathy. I'm going to talk about it a little bit more, but before even saying that, we want to keep away from that dichotomization. Why? Because misfolding proteinopathies are complex, and the prevalent comorbid pathologies strongly influence clinical and pathological phenotype. There's this component of this four horsemen of the apocalypse, A, beta, and tau, which were thought of to be AD specific, synuclein, which is thought to be Parkinson's and DLB specific, and TDP-43. Of course, Dr. Trojanowski had a big role in all of these, but these pictures all actually come from the NIA-AA guidelines because they're all really important for understanding the Alzheimer's phenotype. And like many of the papers that I'm going to discuss, John Trojanowski was a key co-author. But this is data from the University of Kentucky, a project that was the point person was Dr. Erin Abner and her grad student who's now graduated, Dr. Shama Karanth. They looked at people who had mostly been followed since normal cognitive status to dementia. And what they found was that most people were mixed pathologies, and many of them had more than one. The QMP, quadruple misfolded protein, was 19.2%. That had A beta, tau, synuclein, and TDP-43. That coincided exactly with the number of people who had pure AD, just plaques and tangles. So everything that we've been talking about before 1990s, these all would have been lumped in with the plaques and tangles, but they're different. They have a different pathophysiology. And in fact, the most common of them is just late in AD itself. Why is this important? Who cares? Well, if you look at people like we did for long periods of time before death, the average length of yearly follow-up was over 10 years before these people passed away. And there's different ways you can look at cognitive trajectories. In this case, we're looking at years before death and predicted possibilities of becoming impaired. There's different ways you can do it. This is looking at how the disease progresses in the population. The pure late has a relatively slow or mellow trajectory in that sense. Pure AD is worse impairment, but AD plus late is worse than the two pures, and that's really important. But it's sort of confusing at the same time, and so really it gets to what we're all interested in, which is the impact on public health. And so you can assess that question using a fairly simple formula where the morbidity and mortality per person affected of an exposure or a disease or a pathology is multiplied by the prevalence of that pathology or exposure in the population. And there's a metric for that called attributable risk. And here I'm going to shamelessly show somebody else's data, but the question to be asked is what percentage of AD type amnestic dementia in a population is attributable to late NC? And cutting right to it, the late NC is much higher than many people would expect. The implication is approximately 15 to 20% of assigned AD type dementia in this population is attributable to late NC, about 17%. And to put that into context, actually, when we think of AD type dementia, we think of plaques and tangles, but that proportion was less than 40%. So late is 15 to 20, and AD itself is around 40. That gives you some idea of the impact and also the multiplicity of the pathologies in aged brains. And so I just want to send a shout out to colleagues, Dr. Bennett, Dr. Julie Schneider, and the Rush University team. Challenges prior to 2019 was that there was no prior consensus-based effort to tackle TDP-43 in aging, no common terminology. It was very underappreciated and understudied, but there were also opportunities. Classification and terminology could enable autopsy diagnosis. Before this paper came out, over 30% of my cases that I signed out as a neuropathologist could not be signed out. We didn't have a diagnosis. There is no standardized one. And we wanted to also increase awareness and catalyze research in multiple different areas, and ultimately to enable a common language across disciplines. So in 2018 and 2019, there was an NIH-sponsored international multi-institutional group of experts came together, and after 6 months in subcommittees, teleconferences, and then a 2-day in-person meeting, we wrote a report with recommendations. And I would say that Dr. Trojanowski, as usual, had a serious impact on this proceedings. What did we do? We came up with a neuropathology-based diagnosis, classification, and staging that related to the stereotypical progression of the pathology in the brain. And this actually recapitulates and was based on, in fact, a system developed by Dr. Dennis Dixon, Dr. Keith Josephs, and the Mayo Clinic team, another great ADRC center that's made a ton of contributions in this area. So one quick question is how specific the pathology is of late. And one of the key questions is how it interacts and it relates to FTLD. And it turns out that the pathology of late is significantly, not slightly, but significantly different from the pathology of FTLD TDP. This is published in Brain. Again, John Trojanowski was key. This was a great example of a really nice collaboration between two different ADRCs, the more clinic-based ADRC at UPenn and the more community-based ADRC at the University of Kentucky. So late versus FTLD, both have TDP pathology, but there's very different anatomy, different symptoms that are reflected from the different affected anatomy, mostly different ages. And late is far, far, far more prevalent, 100 cases of late for every FTLD. So then there's this evolving appreciation then that John Trojanowski had a big part of understanding the different subtypes of clinical and pathological phenotypes related to TDP, proteinopathy, and aging. There's FTLD, that's mostly younger people with a certain clinical syndromes related to the frontal problems, unusual or the less than 1 in 1,000 lifetime risk. Pure late is much more common, about 100 times more common. It is amnestic presentation. But unfortunately, even more common is the combination of late and Alzheimer's pathology, which is about a 1 in 3 lifetime risk, and is the more severe, swift course of the disease. So follow-up questions related to the pathology is, does late often occur in the absence of either AD or FTD clinical symptoms? And does it eventually progress to FTLD in human beings? The answers to that are yes and no. It does often occur without FTD symptoms or AD pathology. It does not eventually progress to FTLD TDP as a rule. And here are some quotes from Dr. Trojanowski related to some of the stuff that Eddie Lee was saying previously. This is a really nice preparation of some of his quotes in his own words, and also Dr. Virginia Lee's words. When he was asked to name one pathway that's a cause of Alzheimer's disease, he says, well, I think that's erroneous because AD is not one disease. He said, I think that the scientific community appreciates that we should think of Alzheimer's diseases rather than one disease, and the public, I hope, will also appreciate that. There are many forms of this dementia that we call AD. So that gets to the point of what we're actually trying to affect, which is one of the things that John Trojanowski was primarily interested in, is getting this into clinics. How can we get a change of clinical scenarios for people? And that gets to understanding the phenotypes themselves. This is cognition changing over time with no pathology. You typically see no problem with cognition. With pure late, you see diminishment of cognition. That's more severe and fast with pure Alzheimer's disease, but more swift and severe still with the combination of AD and late. The kicker is that these are also the order of their prevalence. Pure late is a very common condition. About 10% of a community-based cohort will have that. However, more common is to have pure AD, and the most prevalent of those three and the most severe is when they're both combined, unfortunately. And so from the phenotypes to be addressed by clinical trials, yes, the future will require treatment for pure late, probably a different treatment for pure AD, but something different, maybe a combination or a third one for the combination of pathologies. So I'm going to sort of get towards the finish by just showing you, of the 60 cases that we've had in the last couple of years, and this was a decreased rate of autopsies because of COVID, but around 2/3 of them had some degree of impairment. This is the NCRAD neuritic amyloid plaques. Around 3/4 of them had some. The absence or presence of any neocortical Lewy bodies, something like 18% had some neocortical Lewy bodies. But in terms of people that had late stage greater than 1, this is a stage of late that is robustly and strongly associated with additive cognitive impairment, taking into account other pathologies. Almost 40% of the people had late stage greater than 1, almost 40%. And this gives you sort of idea of what we're talking about and why this is an urgently needed diagnostic category. So I see that I'm sort of coming near my end here. I just wanted to recapitulate. Late is limbic predominant age-related TDP-43 encephalopathy. It's a common high morbidity brain disease. It's a helpful disease-related concept. It is part of a movement away that was, the charge was, I think, really led by John Trojanowski, away from this dichotomization of dementia as a clinical scenario and dichotomization of pathology as the underlying substrates. And late is completely a different paradigm, and the roles of Dr. Trojanowski were, in fact, many and various. And finally, I'd just like to say thanks to John. I think that what both Dr. Wolk and Dr. Lee said rang very true to me. Dr. John was a mentor for me. He was somebody who taught me through his teachings and his doings. He impressed upon me the importance. He never asked me to help him. He asked me how he could help me. He inspired me by his ability and his desire to not fit in necessarily, but to stick out sometimes, and his ability to have the courage to lead. And I think that As a mentor, those were very helpful to me. As a friend, he would call and we would talk, and he was always, and I learned a lot from him in a lot of different ways. And I think that it's important to note that as the mentor, as you get older and you go from being the job, you're being that of a mentee to that of a mentor. I think that one of the things I think that would make him happy is to know that, in fact, that a lot of his mentees take very seriously that charge, and to take seriously not only the cherishing of the relationship with him, but the cherishing of the relationship to the next generation, and our obligation and taking forth the obligation to make sure that the next generation is strong and smart and able to do things that we can't do. So I'll just conclude that with saying thanks to John. Thanks again to all the organizers and to everyone for putting this together. And that's all I got.
Thank you, Pete. Our next speaker is Adam Boxer from UCSF and will be virtual.
Hi, my name is Adam Boxer from the University of California in San Francisco, and I wanted to thank Oscar for inviting me to speak to you today about tau therapeutics, and also to pay tribute to John Trojanowski, who was a mentor from afar. And I wanted to first apologize for not being able to join you in person at the meeting, but unfortunately I have a family commitment this weekend that I could not miss. So with that, here are my disclosures. What I want to tell you about today is first to just give a little bit of a tribute to John Trojanowski, who was really a pioneer of what we now think of as the field of tau-directed therapeutics. But he was also one of the mentors for my K23 award, and really was very influential in my training early on in my career. After that, I want to tell you a bit about tauopathies, and I think most people here are familiar with tauopathies, but just to review that, talk a bit more about what we've learned recently about tau biology, tell you about tau-directed therapies and clinical trials, and mention how tau biomarkers are particularly useful, we think, for clinical development in Alzheimer's disease. So just a few words to start out about some of the many contributions that John made to neurology and science. And my K Award, which was funded a number of years ago, as you can see, was focused on eye movements. And I was very interested in using ocular motor function to study decision-making in FTD and Alzheimer's disease. And John was one of my co-mentors, and really was always very enthusiastic about this idea because, as he reminded me on a number of occasions, he had studied the anatomy of gaze control in the brainstem, and also had conducted early studies of the anatomy of the visual system using tracing techniques such as horseradish peroxidase and other things. So this was a case report that he published back in 1980 of someone who had selective vertical gaze ophthalmoplegia due to a brainstem stroke in the midbrain. And subsequently, you'll hear that I and he and others are very interested in, particularly in a disease that leads to selective paralysis of downgaze due to tau deposition in the brainstem, and that's PSP. But in our work together from my K Award, we actually did study a number of different neurodegenerative diseases. And this is one of the publications, the early publications that I worked on with John and many others. And here we looked at autopsy-confirmed cases of different neurodegenerative diseases and used infrared oculography, and these are tracings here on the left from normal controls, corticobasal degeneration in PSP patients, and then related the vertical gaze velocity to volume of different brain structures, excuse me. And interestingly, we identified a region very, very close to right here. You can see, hard to see this yellow dot, to what John had identified. You can see 30 years earlier, more than 30 years earlier, from a single lesion study. So, John was very encouraging of this work and wanted to just remember him fondly for that. But what I want to tell you more about are tauopathies and some of the approaches that we think might be helpful to treat these diseases. And most people are familiar with the Brock stages of Alzheimer's disease, where we think of insoluble tau deposition as being strongly related to both the severity of disease, but also the types of symptoms that people experience during life. And we see similar patterns in PSP, Richardson syndrome, chronic traumatic encephalopathy, and other diseases that we call tauopathies. And so it stands to reason that potentially if we could block the accumulation of this insoluble tau, that might be one approach to treating these diseases. And so the general sort of nomenclature and thought of how tauopathies work is that there is a gain of tau function, that is, too much tau is being expressed, it's being chemically modified or misfolded. This somehow leads to neuronal and, in some diseases, glial dysfunction and death. There are thoughts that potentially the misfolded forms of tau can spread transsynaptically in a prion-like fashion from one neuron or glial cell to another, and this may help to explain the progression through the Brock stages and other similar patterns in other tauopathies. And there are cellular mechanisms we know now that can inhibit this toxicity, such as autophagy. So we think about primary tauopathies as diseases where the only protein or the main protein we find in autopsy is tau itself. So tau mutation carriers, PSP and CBD. Secondary tauopathies, the most common tauopathy, Alzheimer's disease, where we think tau accumulates as a secondary to A beta, and also other diseases such as synucleinopathies and even some forms of epilepsy, where genetic evidence strongly implicates tau in disease progression, even though we don't find it deposited at autopsy. So this is a slide about the genetics of tauopathies, and actually the core comes from a review from John Trojanowski's group. And to remind people, this is the tau gene. It's alternatively spliced to give rise to 6 isoforms named for the number of microtubule binding domain repeats, either 3 or 4 repeat tau. In the normal adult brain and in Alzheimer's disease, they're equal ratios, but in certain diseases like CBD and PSP, there's more 4 repeat tau, and we think this may be central to pathogenesis. We also know that there are rare families worldwide where there are point mutations in the gene that give rise to early onset severe forms of dementia, usually frontotemporal dementia. But interestingly, the type of mutation strongly relates to the phenotype, and intron 10 mutations, for example, lead to increased 4R tau production and often produce a phenotype that looks like CBD or PSP. Conversely, when you look at the sporadic diseases and do a GWAS, the strongest genetic risk factors for sporadic PSP or CBD are regulatory regions in or around the tau gene. So this strongly implicates tau as potentially causative in or playing a strong role in the pathogenesis, at least of these primary tauopathies. And of course, we know that these tau mutations have been used to produce preclinical models used for drug development. Well, John Trojanowski and coworkers were really pioneers in thinking about treating tau-related abnormalities, and early in the 2000s thought that perhaps it was tau loss of function. Tau was known to regulate microtubules, and tried to use taxanes like Taxol and apothelone D to make up for tau loss of function. And these are some of the publications. And there was good evidence from animal models that this might actually work. And so I was very inspired by this, but had a hard time getting a hold of one of these brain-soluble taxanes initially. And so we started working with a different, what was purported to be microtubule stabilizing agent called davunitide. I will say that John and Virginia were not happy with me about this because they thought the science behind this microtubule stabilizer wasn't very strong. But in any case, we were able to do a large international clinical trial in PSP. The drug had no effect over treatment over a year, but this was an important study for PSP clinical trials because it helped us to demonstrate that these large international trials are feasible and you can get a clear answer in a year. Eventually, and I will say that John and others, and we all tried to get brain soluble, brain permeable taxanes, and eventually we were successful in getting a taxane called aviotaxane, and we tried testing it in 3 different tauopathies: Alzheimer's disease, amyloid negative corticobasal syndrome, and PSP, and we used biomarkers. Again, John played a big role in the CSF biomarker development to convince ourselves that we could premorbidly accurately diagnose these different tauopathies. But unfortunately, what we found is that we had to stop the trial early because the drug was too toxic in Alzheimer's patients to go to the highest dose. And in the PSP and CBD patients, there was a dose-related worsening in dementia measured by CDR sum of boxes, although we did find a small but significant effect on reduction in CSF inflammation as measured by YKL-40. And so that is the last of the clinical trials of microtubule stabilizers thought to make up for loss of tau function, although at least that I'm aware of. And now really, based on a better understanding of tau biology over the past few years, we now know that tau may really, there's a lot of evidence that gain of function, either through misregulation of synaptic proteins and synaptic vesicles, or interactions with the mitochondrial proteins, or even through interactions with nucleic acids, may actually play important roles in neurodegeneration. And it may not be just the loss of microtubule regulation by abnormal tau that's important in disease. And so this gain of function hypothesis really has taken over in tau therapeutics. It's also based on work from Leonard Mucke and co-workers, as well as others, suggesting that, for instance, in an APP transgenic mouse that overexpresses amyloid, about 15% of these mice will die after 6 months. But Leonard and Eric Roberson showed that by genetically knocking down the amount of tau protein in the brain, these mice become resistant to this toxicity. Not only that, but if you knock down tau protein genetically, mice become resistant to other forms of toxins such as kainate, which produces tonic-clonic seizures. Well, these are exciting times for tauopathy therapeutics because there are now antisense oligonucleotide trials. Here's an example of one that was presented last year at AAIC, where you can see that with an antisense oligonucleotide, you can knock down tau levels in CSF by a similar amount as in these mice. And so far, it seems to be safe and well tolerated in human Alzheimer's patients, although we don't yet know whether this is clinically producing a benefit. If it does, it would be a very nice confirmation of this hypothesis that tau is central to neurodegeneration. And I should mention, there are also ASO trials going on in PSP. What other types of therapies are targeting tau? Well, again, this is what we think. This is how we think tau may lead to toxicity and neurodegeneration. So initially, it's produced as a normal protein, but there may be post-translational modifications that lead to toxic forms of tau protein that aggregate. If these are not cleared by systems in the cell, such as autophagy or the proteasome, they may spread to other neurons or glia, where they may template other aggregated forms of tau, which again, if they're not cleared, may lead to toxicity, and again, may form even larger aggregates that we eventually see at autopsy. So I mentioned that there are antisense oligonucleotide therapies and eventually gene therapies targeting genetically targeting the production of tau. There are small molecule enzyme inhibitors in the clinic that, for instance, kinase inhibitors. There are aggregation blockers, and there are new drugs that may enhance clearance, for instance, through the proteasome of tau. Of course, the most advanced therapies in clinical trials are immunotherapies, but unfortunately, they haven't been particularly successful so far. And one clue that we're getting from recent really amazing science, and this is cryo-electron microscopy that has been able to solve the three-dimensional structures of different tau protein aggregates in different neurodegenerative diseases, is that we now understand that the toxic, or what we think is the toxic core of the tau molecule in the aggregates, is really in the microtubule binding domain in the C-terminus. And in different tauopathies, it turns out that the three-dimensional structure of this core is different. So it's different in Alzheimer's disease and PSP. Well, I mentioned that the initial tau immunotherapy trials have all been negative. Here's an example of 3 of the antibodies, 2 of which didn't work either in PSP or Alzheimer's disease. But it may be explained that these molecules were really targeting the N-terminus, which was far away from and even often cleaved off in these toxic aggregates. And so newer antibodies and trials and newer CSF biomarkers, which may be more informative, are really targeting this aggregated core structure. But it raises an important question. If the core structure is different in different tauopathies, then will we need different immunotherapies because they may bind differentially to these different 3-dimensional structures? And I think this question remains to be answered. And so I think John might possibly agree with me or have agreed with me that it's important, therefore, to think about testing these different tau therapies in different tauopathies. And so Alzheimer's disease is the most common, but really genetically is the least related to tau protein. But as I'll show you in a minute, they're really great biomarkers that we can use for clinical development. PSP is strongly related genetically, but is rarer. And we don't have very good biomarkers. And of course, we'd love to do trials in tau mutation carriers because these are like human transgenic mice, but they're very rare. And we are making efforts to generate an international, global cohort to prepare for clinical trials, but I don't think we're quite ready yet. And so again, it's the Alzheimer's cases that really have the inside track in clinical development, and that's really because of tau biomarkers. So what do I mean by that? Well, again, here's my schematic of how I think tau may lead to toxicity in cells. And we have very good biomarkers in Alzheimer's disease, but not other tauopathies. And so we can detect insoluble tau in the brain using PET. We also know that when tau gets out of the cell, it's often cleaved into fragments. These fragments can make it into the cerebrospinal fluid, and most of them are N-terminal fragments, and these are the typical CSF tau assays, p-tau 181, for instance, and total tau. And but now, which I won't have time to talk about, with the knowledge of the new 3-dimensional structures, there are even newer assays using mass spectrometry that may even be able to detect the microtubule binding domains. But of course, what's probably most exciting in Alzheimer's disease, at least from my perspective, is the idea that some of these fragments even make it into the blood and can be used diagnostically, potentially in symptomatic people, to diagnose Alzheimer's disease. Well, why is this so exciting for development of therapeutics? Well, some of the work that really at least excited me in Alzheimer's disease was the phase 2 trial of donanemab, an anti-amyloid treatment, which is now in phase 3. And this was actually a positive clinical trial because the primary endpoint, this clinical composite, the IADRS, what the decline was slowed significantly in the group that was treated with donanemab. And what you see is that there was a strong parallel in 2 biomarkers of tau. So both the patients who responded had less insoluble tau protein that accumulated in the frontal lobe. So here's the placebo group, here's the treated group. But even more excitingly, this blood biomarker of tau, plasma p-tau 217, was strongly reduced in the patients who were treated with drug as compared to the placebo group. And so this raises the possibility that we could start to use these biomarkers in early phase, possibly biomarker proof of concept trials to de-risk other therapies. And really, you would think that if you have an effective tau therapy, you might really particularly expect to see these effects on tau biomarkers. But one of the sort of remaining questions is that we know that donanemab was not a tau therapy. It actually is very effective at reducing amyloid. And so how much of what we measure in the fluid phase and in the soluble phase is directly related to tau toxicity and the role of tau in neurodegeneration, and how much of it reflects an intermediate step related to amyloid toxicity in Alzheimer's disease. And I think this is work from Oscar Hansen just suggesting that particularly the fluid biomarker plasma p-tau may mediate the changes that we see in insoluble tau by PET. And so I think as we think about developing new therapeutics, particularly tau therapeutics, this is an important question that we need to resolve, and I think something that really we can resolve and should resolve in the setting of clinical trials. So with that, I want to just thank you for your attention today, and again, apologize that I couldn't be there in person, but I'm really grateful to Oscar for allowing me to present and to really present my fond memories of John, who was really a wonderful mentor from afar and someone whom I really enjoyed working out with in the gym when we were at meetings together. So what I told you is that the tau protein is strongly implicated in many neurological diseases, and we think that we should be investigating new tau therapeutics in all of these different tauopathies. That reducing tau gain of function is the current leading therapeutic strategy, and there are a variety of ways we're doing this. John and coworkers really led the field of tau therapeutics by trying to make up for loss of function, but those therapies have really, I think, fallen somewhat out of favor. We think that Alzheimer's disease really has the inside track for clinical development of therapeutics, and that's because of the availability of blood tests. And I think that the blood biomarker p-tau217 and other related p-taus in the blood may be particularly useful once we truly understand what they mean and how they reflect amyloid versus tau deposition. But unfortunately, so far, no successful tau therapies. I think, again, work with donanemab and evidence from other studies suggests that potentially if we can, in early phase trials, see less tau deposition using tau biomarkers like PET and maybe hopefully even plasma p-tau, this might help us to predict which drugs should go on to larger clinical trials that are powered to look for clinical effects. And so this is what we're hoping to do in the future. I would say we're nowhere near ready to give up on tau therapeutics. Really, we've only barely scratched the surface on the first generation of therapeutics. And with the amazing new insights into tau biology that we're getting, for instance, the three-dimensional structure of tau, we're really able to develop new biomarkers and therapies, and really will ease our trials, not just for Alzheimer's, but also for PSP and tau mutation carriers. And just wanted again to finally thank John for his mentoring and for really his work in pioneering and starting, this field of tau therapeutics. And thank you for your attention, and just wanted to thank all my fantastic colleagues at UCSF and the Memory and Aging Center, and particularly acknowledge support from the National Institute of Aging, the Tau Consortium, and NINDS for all this work. Thank you.
Thank you, Adam. Our next presenter is Cherise Watson. She's coming. I noticed that there is an overrepresentation of the UC system in the speakers today, so we need to fix that problem.
All right, good afternoon, everyone. Thank you so much for allowing me to present my research today. So my name is Cherise Winston, and I'll be talking about the work that we've been doing at UC San Diego with blood-based biomarkers. So many of us in this room are well aware how important it is to diagnose Alzheimer's disease at early and more treatable time points. The current state of AD diagnostics are expensive, they're time-consuming, and they're frankly invasive. So many of us in the field have been focusing on the importance of looking at biomarkers in our blood. And so what we have been focusing on is assessing this from the perspective of exosomes. So what are exosomes? Exosomes are small, nano-sized microvesicles that are known to be secreted from a variety of different cell types, including neurons, as well as glial cells. The function of these microvesicles are not well understood. They have been shown to participate in short-range intracellular communication, as well as been thought to participate as an alternate waste removal mechanism from the cell. What is notable about exosomes is primarily the cargo in which they contain. They're known to contain a diverse set of molecules, including lipids, proteins, microRNAs, and a host of other molecules. What us in the field have been focusing on has been characterizing changes in exosome cargo levels in hopes that they can serve as diagnostic markers for identifying different stages in Alzheimer's disease. Before we get into some of the data that we have generated in our lab, as well as speak about the collaboration, one of our most recent collaborations that we established with Dr. John Trojanowski before he passed, I wanted to make sure I highlighted the methods in which we use in order to isolate these exosomes from human blood. So what we use is a commercially available kit. We precipitate our exosomes using a polyethylene glycol-based method, and that kit is from System Biosciences. And then the next thing that we do is that we generate what I call a BAE complex, a bead antibody exosome complex. Our magnetic bead is covalently linked to streptavidin, and with that we can use any biotinylated antibody in order to isolate different subpopulations of our exosomes. We have, the bulk of our work has primarily been looking at neuronally derived exosomes, and we have been using L1-CAM or CD181 to isolate that specific subpopulation. We've also become very, very interested in looking at other exosomes derived from other cell types. So we've used GLAS, which is a glutamate transporter, and that is going to be localized in mature astrocytes. And then we've also been focusing on TMIM119, and that is a marker that we've been used to identify exosomes that are derived from microglial cells. After we have generated our BAE complexes, we can utilize a ExoFITSI secondary marker that's going to ubiquitously bind to all exosomes, and then we can fact sort them based off of fluorescent intensity. So after we have generated our BAE complexes, we can characterize our exosome preparations, and what's seen on the screen here are representative FACS plots that we can generate through our characterization. So what we can see here that based on fluorescent intensity, there's a significant degree in flow separation from our non-exosome negative control, and so this just confirms that FACS sorting is an effective method in isolating our different subpopulations of exosomes. Other ways in which we can characterize our exosome preparations are by size. Exosomes, these small specific class of microvesicles, are between 80 and 100 nanometers in size, and so we can use nanotracking particle analysis, or NTA, in order to characterize our exosomes based off of size. Our methods also allow us to generate equal concentrations across our different patient groups, and that together allows us to accurately quantify changes in exosome cargo levels as in relation to disease state. Okay. So this was one of our earlier studies that we had done, again, characterizing exosome cargo levels from our 4 clinically distinct patient groups. We have patients that were identified as cognitively normal controls. We have autopsy-confirmed AD patients, and then we also were able to identify a sub-cohort of MCI patients and a secondary cohort that declined to AD-like phenotype within 36 months of their initial diagnosis. What you're seeing in the first panel here is CD81. That is another method in which we use to characterize our exosome preparations. CD81 is a traditional exosome marker, and we utilize that to normalize against all of our exosome cargo levels. Well, we can see here in this earlier study that we saw a significant increase in the amount of amyloid as well as 2 different epitopes of tau that are contained in the neuronally derived exosomes that are taken from our more demented-like patients. So we were very excited about this work, and that is what actually led to us to investigate more intensely about the potential the biomarker potential of our exosomes. So in our first study, we were able to conclude that plasma neuronally derived exosomes can accurately differentiate AD patients from control, and then based off of the changes that we were seeing in our two subcohort of MCI patients, we were also able to see that changes in exosome cargo levels could actually have a predictive ability in predicting the conversion from MCI to AD. We've also looked at exosomes as potential disease monitoring tools. In this interesting study, we actually quantified changes in different synaptic proteins and neuronally derived exosomes that we had isolated from MCI patients, and these patients were on a growth hormone treatment for a specific period of time. We did see an overall reduction in a number of synaptic proteins in these MCI patients. However, it was independent of this growth hormone treatment. And the last study that I wanted to highlight is that, as I said, we've become very interested in assessing the biomarker utility of exosome cargo that are derived from different subpopulations. So in our last study, this was a collaboration with the VA, was that we were able to isolate neuronal as well as astrocyte exosomes from the same patient, and we quantified and specifically levels of amyloid beta and the neurogranin, which is a postsynaptic marker. We were able to show that changes in these cargo levels were able to differentiate a military personnel cohort, our TBI patients who self-reported sustaining between 1 and 3 MTBI impacts upon their return from military deployment. And that was a potential diagnostic marker from differentiating that patient group from our control patients who had not sustained a head impact. So again, as we are constantly and become very, very interested in continuing to assess the biomarker utility of our exosomes and see if it can be applicable to other neurodegenerative diseases, that is what actually led us to form our collaboration with the late Dr. John Trojanowski. So we became very, very interested in assessing how TDP-43 could actually serve as a useful biomarker, and this was specifically in the context of late. So we've already heard a little bit about that, so I won't go too much into detail about that, but I did want to highlight where this kind of connection came from. So TDP-43 pathology, which is one of the pathological hallmarks of late, has also been reported in other neurodegenerative diseases, including AD, ALS, as well as frontotemporal dementia. The propagation of TDP-43 has also been reported to be mediated through exosomes. So we thought that it was important to assess that if we could not only detect TDP-43 levels in our exosomes that were derived from blood, and if they could actually serve as a potential diagnostic biomarker for patients who were diagnosed with late. And I wanted to also highlight an interesting point of the overall impact of this study, as we all know, is that late is a comorbid condition, a recently described condition that is difficult to diagnose in living patients, and currently the only way we can diagnose late is post-mortem. So that also is what drove our interest, was to see if we could potentially identify a biomarker that could diagnose this condition in living patients. So this is a representative of the demographic of the samples that we included in our study. These samples were gifted to us by Dr. Pete Nelson, so he already described the importance of late and his contribution, as well as Dr. Trojanowski's contribution to this field. What you can see here, a relatively small cohort that we understand, but we do have autopsy-confirmed late patients in our sample. And we thought it was important to compare our data to a mixed clinical cohort of patients. We really wanted to see if the accumulation of TDP-43 in our blood-based exosomes, if they could truly have powerful diagnostic ability in identifying patients who are already identified with late. So our Dr. Trojanowski was gracious enough in order to donate or to give us some antibodies that we could utilize in our studies. So at the time that we had began this investigation, there were not a lot of assays that were available or suitable for us to measure TDP-43 in our exosomes, and so we decided to generate our own in-house ELISA. And so his generous contribution allowed us to create an ELISA, and it directly led to the data that I'll show you in the next slide. And so what's represented on the slide here are just a number of the different antibodies that we used to optimize for our assay, and the one that was highlighted here is the one that we actually ended up using for the subsequent data. So on this slide here, it's just an overall schematic of our workflow, how we generated our in-house ELISA. On one side, you'll see that we have our just traditional exosome isolation methods, our purification methods that we did to extract these exosomes from our human blood. And what was also notable and most interesting about this study was that this was actually one of our first studies in which we were assessing exosome cargo levels, specifically TDP-43, from 3 different subpopulations. population. So we were assessing TDP43 levels from our neuronal, our astrocyte, as well as microglial-derived exosomes. And again, they're coming from the same patient. In order to generate our in-house ELISA, we developed a sandwich-based ELISA. We utilized the TDP43 antibody that was gifted to us as our coding antibody, and then we used a recombinant full-length antibody as our sandwich ELISA for detection. Excuse me. Okay. So what's shown here again is just our ability to characterize our exosome preparations from human blood. And so again, a significant degree in flow separation indicates that we can utilize this process effectively to generate our 3 different subpopulations of exosomes. And then, as always, we use CD81 to normalize all of our exosome cargo levels in order to ensure that the changes that we see are a direct relation from the exosome that is released from the TDP43 that is released from our exosome levels. Okay, so as I said, we were very interested in developing our own in-house ELISA, but then we also were able to find a commercially available kit, and we thought it was important to compare our data that we had generated from this kit. And so I'll walk you through the 3 different data that we acquired from our 3 different exosome populations. So what you can see here on the first panel is that these are exosome cargo levels of TDP43 that were taken from our neuronally derived exosomes. What we can see here that we are actually able to measure TDP43 in our exosomes derived from our autopsy-confirmed late patients and compared to our mixed clinical cohort. However, we can also see that we don't see any significant difference between the 2 groups, suggesting that TDP43 does not have sufficient diagnostic capacity to differentiate these 2 groups. What was most interesting was that when we assessed TDP43 accumulation in our astrocyte-derived exosomes, we were able to see a significant increase in the amount of TDP43 accumulation in our autopsy-confirmed late patients in comparison to, again, our mixed clinical cohort. And what was also convincing to us was that this was consistent across our in-house ELISA as well as the commercially available kit. Another thing that was also very interesting for us was the quantification of TDP43 in our microglial-derived exosomes. So as you can see here at the top panel, the commercially available kit, we're detecting a significant reduction in the amount of TDP43 in our autopsy-confirmed lay patients. However, in our in-house ELISA, we actually seen a significant increase in the amount of TDP43. And so this is something that is of great interest to us, something that we were still trying to assess and determine what could be the potential reason for that. As I said, the antibodies that we were graciously gifted from Dr. John Trojanowski, I believe could be the major driving factor for this. The antibodies that we're using are specifically localized to the C-terminal region and instead of using a full-length TDP antibody. So that particularly could be driving the differences that we're seeing here. But nonetheless, we were very, very excited to see that specifically in this subcohort of exosomes, our astrocyte-derived exosomes, that in significant increased levels of TDP43 could actually be a potential biomarker for this particular disease. What we were also interested in determining is if any demographic or cognition-based variables influenced or had any kind of correlation with the accumulation of TDP43 that we determined in our 3 different exosome populations. And what we can see here when we assess changes in MMSE score or when we assess changes in sex, that across all 3 of our different exosome populations, that we were not seeing any changes in exosomal TDP43 in relation to these variables. We also wanted to assess if ApoE genotype had any influence in the exosome cargo levels that we were quantifying, again, in our 3 different subpopulations. And again, we're not seeing any changes in exosome cargo levels in comparison to ApoE genotype. So this further suggests to us that none of these variables are driving that increased exosomal level that we're seeing specifically in our astrocyte-derived exosomes. And again, thus confirming that this could actually be a potential potential biomarker for patients who have late. Lastly, we thought it was very, very important to see if we could measure TDP-43 in plasma. This is a unique situation with the samples that we had. So these are exosome-depleted plasma. After we precipitate our exosomes, we keep the plasma supernate, and we were able to measure TDP-43 again in our autopsy-confirmed late patients, as well as our mixed clinical cohort, but we're not seeing any changes, any significant differences in the levels of TDP-43 that we measured in our in-house ELISA as compared to the commercially available kit. This is also something that we're very, very interested in looking further into, specifically if we can measure TDP-43 in straight plasma that has not been exosome depleted. Okay. And so I just want to summarize the work that I have presented here. TDP-43 can be measured in our 3 different exosome subgroups, in our neuronal, our astrocyte, as well as our microglial-derived exosomes. We are seeing a significant increase in the amount of TDP-43, specifically in our astrocyte-derived exosomes, suggesting that this could be a novel and potential blood-based biomarker for those who are diagnosed with late. And something that has been mentioned quite a bit, or at least earlier on in this meeting, is that one of the future directions and where I'd like to see this project going is we really want to assess how race and ethnicity is going to influence the diagnostic capacity, not just of TDP-43 as a biomarker for late, but all the diagnostic measures that we've done thus far in relation to Alzheimer's disease as well as other neurodegenerative diseases. It's something that I know that has been lacking in this field, and I know there's a significant effort in order to kind of rectify that, but I think it's going to be very, very important as we move forward in looking at blood-based biomarkers that we assess that as a context of this diagnostic capacity for exosomes. So with that, I would like to thank all my collaborators, my mentor, Dr. Rissman, and again, this work would not have been able to move forward if it wasn't for our collaboration with Dr. Pete Nelson, as well as with the generosity of Dr. John Trojanowski. So thank you.
I'm not sure if we have time for questions. We have one there. We have 9 minutes.
Yeah, thank you very much, Possin, University of Michigan. Your astrocytic-derived exosomes seem to have a fair amount of scatter in terms of the amount of TDP-43 you see there. Have you had a chance to go back and look at the neuropathology in those cases that you have to see if that tracks with the degree of astrocytic pathology that's there? Thank you.
Okay, if we don't have more questions, I would like to thank all the presenters. Were excellent. And I hope that we can meet in person in the fall of 2022 and without using a mask. Thank you. Bye.